Hello, everyone. We're going to wait a minute here before we get started. Okay, I see people are still showing up. Ah, the number has stopped. Okay, so I will just start. Hopefully some people will trickle in as we go. So welcome to the second day of the virtual SOLAS Summer School. Glad to see you all here. Today will be about the SOLAS core theme 2, air-sea interface and fluxes of mass and energy. But before we start with our plenary lectures, I have a few things to remind everyone. The first is please, if you feel comfortable, I highly encourage you to turn on your cameras, at least during the discussion time, so that we can get a sense of each other and really feel like we're interacting. So just a wish from our side. The second is that, we have the recording from yesterday already uploaded to Whova, so everyone is encouraged to check out the recording if they missed the session yesterday or if they want to revisit the session from yesterday. And all of these things are on Whova for the next 3 months, I believe. Yes, 3 months. So please use it as a resource. And also please use Whova. I just want to remind everyone again that you can really talk with each other, ask the lecturers questions all throughout on Whova. And the final thing is that we have these competitions going on, so please don't forget to enter into the competitions, one for poster creativity per day, one for content per day of the posters, and then one general prize for photos and one general prize for the best, or so for social media posts. So using the hashtag SOLASSchool. Finally, just remember that if you have technical issues, our colleagues from Mindfully Wired are here in the background, Becky and Paul, to help. So if you have any issues, please, please write to them in the chat. Okay, so without any further talking from me, I'd like to introduce our lecturers for the plenary lecture. So as I said, it is the core theme 2, so air-sea interface and fluxes of mass and energy. And like yesterday, we have first an introduction on the topic, then we have a research highlight, and then a lecture on a related cross-cutting theme. So the first lecturer is Tom Bell from Plymouth Marine Laboratory, and he will be talking about the air-sea interface and fluxes of mass and energy, giving an introduction. So with that, I'd like to hand it over to Tom.
Okay, thank you. So, yeah. I'm Tom Bell, Plymouth Marine Laboratory in the UK. I'm going to be talking about the air-sea interface and fluxes of mass and energy, or as I have been known to call it, what the flux. There's a lot to cover in this 20 minutes, so I will do my best. First off, I thought it sensible to start with a definition of flux, which is the transfer of a material or energy over a certain unit area per a certain amount of time. And I've got a number of equations here on the left-hand side of your slide. First two focusing on transfer of heat. These are bulk equations, sensible heat, which is the direct heat transfer, or latent heat, which is the transfer of heat due to the evaporation of water. And then there's a number of terms in there, but draw your attention particularly to the delta T and delta Q terms, which is the difference in the temperature or the water vapor mixing ratio between the air and the sea. And you also have the transfer of momentum from the atmosphere into the ocean generally, which has the density of air in there. And then finally, gases, where the flux of a gas is a function of the delta C, which is the gas concentration difference. And then the red K's that are all highlighted in each equation, they're not always presented as a K, but I've kept them all consistent. The point I would make about that is that they are all transfer coefficients that in general for the heat and the latent heat and the momentum are a wind speed dependent term. Of course, the title includes aerosols and particles. I'm not going to get into that, I don't have time, but there will be some of that in the next talk. So, starting off with a sort of conceptual view of gas transfer, we have here the horizontal blue line is the air-sea interface, and then the vertical lines represent the concentration. And in this example, we have a higher concentration in the air, the bulk air concentration, than in the seawater. And then at the interface, or either side of the interface, is what's termed a diffusive sublayer. And it is the transfer of the gas across this diffusive sublayer there's a resistance to that transfer, which is represented on the left with R total. Now, another way to think about that is with the piston or syringe that I've drawn on the right-hand side, where the plunger is creating a delta C, it's creating a gradient, and then the resistance is, in this analogy, is the diameter of the syringe or the tube that limits how fast that concentration or that pressure can result in a flux into the ocean. So that's how we tend to think about it. This is work that was done by Peter Liss and Slater in 1974. And when you represent that mathematically, the flux is a function of the delta C divided by the resistance to the transfer across that diffusive layer. But actually, when we talk about gas transfer, we tend to talk about K, as I mentioned before. And K is the gas transfer velocity, or sometimes it's been referred to as the piston velocity, and it's equal to the inverse of the resistance to transfer. And so typically you will see equations that write that the flux is a product of the gas transfer velocity multiplied by the concentration difference across the air-sea interface. Finally, just to note that you can actually then divide that resistance between the air side resistance and the water side resistance, and I'll come to that in a minute. So, another point of clarification about this model, you may well see, and particularly if you work on insoluble gases such as carbon dioxide, a slightly simplified or modified version of the model, where because the gas is insoluble, all of the resistance to transfer is actually on the water side of the interface, meaning that the resistance in the water is equivalent to the total resistance. And this then means that it's the same idea. The flux is the delta C divided by the resistance in the water, and then the flux is equal to the K, the small k, lowercase k, W times the delta C. But that's not the case for every single gas. Okay, but at the moment I've just talked generally about gases. I haven't specified different gases, and of course they have different properties. And one of those particularly important properties is the diffusivity of the gas. And on the right-hand side of the slide, you can see that I've got diffusivity on the y-axis and temperature on the x-axis. And different gases occupy very different spaces in terms of their diffusivity. The helium is very, very diffusive compared to DMS or SF6. And then all of these gases have a dependence on temperature. So as the water gets warmer, the gas diffuses faster. But we represent the diffusivity in the gas transfer, not just in terms of diffusivity, but also in terms of the properties of the water itself, so the viscosity of the water. So you can think of that with these two pictures of swimmers. If you imagine that these two guys can swim at the same speed, I'm not sure they actually can, but if you imagine they can, they're swimming through very different media. The one swimming through the muddy water, that water is more viscous, it's harder to swim through. So that will affect the Schmidt number, which is this property that describes both the seawater and the gas. Whereas the guy swimming through the clean water can swim faster. And we use this Schmidt number to scale our gas transfer velocities to account for the effects of temperature and the properties of the gases. So we take the ratio of the Schmidt numbers raised to an exponent. I won't get into the discussions about how that exponent of a minus 0.5 comes about. And we use that to scale our gas transfer velocities from one gas to another, or for different environmental properties from one environment to another. The other very important property is the solubility of the gas, which is often referred to as Henry's law. If you look at the table on the top left, that represents the dimensionless solubility, which is the water concentration divided by the atmospheric concentration at a fixed temperature. And you can see that methanol is much more soluble than CO2. The ratios are tremendously different. So different gases have different solubilities, and then each gas has a dependence on the temperature. So on the right-hand plot, we have solubility on the y-axis and temperature on the x-axis, and you can see that as the water warms, it can hold less gas, and the solubility decreases. This is an example for DMS. So we use the solubility in the flux equation in two ways. One is in how it influences the gas transfer velocity. So I've written the gas transfer velocities here as reciprocals, which is equivalent to the resistance. And you can see that the solubility term is there divided by the air side gas transfer velocity. And that's why something like CO2 with a very low solubility, the air side is not particularly important in identifying the gas transfer velocity, the total gas transfer velocity. We also use the solubility in order to calculate the delta C, because you need to have your concentrations in the same units. So in this example here, the air side concentration is being converted into water side units in order to get a delta C in water side units. Now, the calculation presented here is a simplified view that is more complicated when it comes to CO2, and I encourage you to check out a paper by Wolf et al. For a fuller explanation. Oh, where are we? Jumped ahead. Okay. Now, what I presented so far is all conceptual, and of course, the real world is far more complicated than that. So KW really, the gas transfer velocity, is really a bulk term representing all of the processes that can restrict transfer across the interface. And there's a whole range of processes. The ocean is full of biology producing other compounds that can affect the properties of the surface layer, that can interact with the chemistry. You can obviously got the sunlit zone, so it's photochemistry, and then you have deposition of particles and rain onto the surface, all of which can influence gas transfer. And this all occurs at a range of spatial scales. And yeah. So I've listed a whole range of different processes on the top left here that are known to influence gas transfer and will have an impact. And these two images, I think, nicely encapsulate how different the ocean can be at different spatial scales. And these processes are not only relevant for an individual gas, but they will also potentially have different impacts on different gases to differing degrees. So the grand challenge, if you like, is to try and estimate and understand and parameterize the gas transfer velocity in order to make good flux predictions of a range of different gases. And I'll spend the next part of the talk talking about different approaches that have been used to try to understand the gas transfer velocity. These can be broadly separated into sort of lab and tank experiments, budget techniques, and micrometeorological techniques. And the point I really want to emphasize is that I'm going to talk about a few of these very briefly, but they operate on very different temporal and spatial scales. And this is important when it comes to understanding what those results mean and how they can be interpreted and indeed how they compare with each other. So firstly, briefly about wind wave tank experiments. In general, this is an approach taken where you have a tank full of water and you blow wind across the surface like the video shows in the bottom right. They can do various things where they introduce waves but with a paddle pushing the water, or indeed in the top left you see the Aeolotron, which is a state-of-the-art tank in Germany, where it's a circular flow of wind and indeed water in order to try and accommodate challenges with the fetch of the wind. And some of these results were first used by Peter Liss and Lehahn in 1984 and produced a broken set of linear equations to parameterize the gas transfer velocity. And you're going to see a lot of these parameterized against wind speed because it's the primary physical forcing in the ocean, where they argued that there were 3 different regimes in the ocean: a smooth regime, followed by a rougher regime where there's some shear stress from the wind, and then finally a breaking wave regime where there are waves and bubbles. Following on from that, so of course the thing to consider with wind wave tanks is that they are of course not the real world, and so they can tell us something, but they are potentially limited in terms of maybe the fetch or the depth of the tank, and thus the sort of dynamics you get between the wind and the waves, and how representative they are of the real environment. And many of the tanks use fresh water because they want to preserve the longevity of the tank. But of course, that leads to different properties in the water that need to be adjusted for. Okay, so the next approach is a budgetary approach at a global level, using excess radioactive carbon from the release due to bomb testing. And this was used to do a budget and look at what the C14 in the ocean was. And from that, you can get an average gas transfer velocity at the average wind speed for the ocean, over the ocean. And Rick Wanikoff has done a huge amount of work on this, and his most recent publication in 2014 is a non-linear parameterization that fits through that carbon-14 estimate. So that's great, different to the lit wind wave experiments. But of course, this is limited because it is only one data point. And so from that, you have a very large spatial and temporal scale. And so it doesn't necessarily reflect the variability that you might see over regional, different parts of the ocean, or indeed interannual variability. The dual tracer approach was pioneered by Andy Watson and others early in the late 20th century. Uses 2 different gases that are very similar in their solubility, but very different in their diffusivity, their Schmidt number. And broadly, the approach here has been to go into the ocean. This is an example from just New Zealand, and to release a large amount of these gases into the water and to track them. And you assume that the mixing of the gases, or mixing of the tracer, affects both of the gases equally, and that the differences between the 2 concentrations is related by the Schmidt number. And from that, you can derive gas transfer velocities. Now, there are challenges with this approach in terms of keeping track of the patch and making estimates of what the mixed layer depth is. And there are also some limitations. The time scale is still relatively long, many hours to days. You get a few data points per cruise, and there are only a few data at high wind speeds. And these gases are particularly insoluble, so how relevant they are, the results are to more soluble gases is a question. Here's the gas transfer velocity against wind speed plot again. The Lehahn line is plotted for reference, and you can see all the dual tracer results that have been collected. This has really helped to start to constrain the relationship with wind speed. Although note that there are only 3 data points above about 12 meters per second. And just to emphasize again that this does cover the range from the smooth regime to a rougher surface and onto breaking waves. Finally, we have micrometeorological techniques. There are a number of these, but I'm going to focus on eddy covariance, or EC for short. Now, the principle of this, we have our sensors as a tower imagined over the ocean with the wind moving from left to right. It's the big pink arrow. And of course, horizontal wind contains a lot of turbulent eddies. And the principle of the approach is that the eddy that I've highlighted with the yellow and orange colors, as the wind or the eddy moves towards the sea surface, the flux is coming out of the sea surface, so that eddy contains lower, on average, concentrations of the gas of interest, and then it picks up the gas coming out of the ocean, and then as it returns away from the sea surface, it has a higher concentration. And so if you look at the fluctuations in the concentration along with the fluctuations in the vertical winds, you can derive the vertical flux. And this very powerful technique, it gives an opportunity to look at new and different gases. And if you combine the flux measurements with measurements of delta C, as the equation on the bottom right shows, you can then derive the gas transfer velocity. I haven't got time to talk about it in a lot of detail, but if anybody's thinking about doing it, particularly for CO2, then I really urge you to avoid contamination of your signal by using a closed path instrument and drying the airstream. We can talk about that more later if people want to. So, of course, the eddy covariance technique didn't immediately work. There were a number of problems, some of them related to my plea for a closed path dried gas stream. So initially the results from eddy covariance were on orders of magnitude too high, and there were challenges with making the observations over the ocean. But more recent estimates are now much more in line with the observations that have come from the dual tracer approach. And so this plot here is wind speed against the gas transfer velocity, and it's a collection of a number of the different observations that have been collected for CO2. And the grey line is the dual tracer gas transfer velocity parameterization. Of course, that doesn't mean eddy covariance is a perfect technique. It does require instruments that can operate at high frequency, and it doesn't always have sufficient signal to noise to make it a reliable observation, and it's challenged at very low wind speeds when the eddies become too large to observe easily. And it's relatively imprecise. There's still some averaging that needs to take place on the order of a few hours potentially. And that, of course, then means you're averaging over space as well as time if you're on a moving ship. So I think this is pretty much my final slide. Just a note of some of the challenges we still have remaining. I've shown again the gas transfer velocity against wind speed plot, but I've added the gas transfer velocity parameterization that you get approximately when you look at dimethyl sulfide. Now, dimethyl sulfide is not as insoluble as CO2, so we expect that to plot below the various lines that are the CO2 gas transfer velocities. But CO2 is more soluble than the dual tracer, the helium and the SO6, and yet they seem to agree with each other. And these differences between the DMS and the dual tracer and the CO2 are greater at high wind speeds, and the current school of thought is that may be driven by the role of bubbles. But if that was the case, why is the CO2 not less than the dual tracer? So we still have work to do to try and understand some of these processes that we think are important. Okay, so just to sum up, the concept that drives the observation of gas transfer is the thin film model, which is not a stagnant film. There is renewal and flushing of that thin film. It's a nice simple concept that we use to characterize gas exchange. And the gas transfer velocity is thus a bulk parameter that covers all the relevant processes. As I've gone through a range of the approaches used for studying and constraining gas exchange. And fundamentally, wind speed is important and explains a large amount of the gas transfer over the ocean. When we're now starting to really look at the different processes that might cause some of the variance between gases or in different conditions. The eddy covariance technique has enabled the study of some of these processes over shorter timescales, but we have a number of remaining challenges and uncertainties. And as I mentioned, bubbles and waves are one of those. What's happening at extremely high winds is very unknown. And then, as will be covered in some of the following lectures, the processes in the microlayer and the influence of surfactants and the influence of partial ice cover pose challenges for the future, as well as what's going on in coastal shallow waters and when it rains. So with that, thank you very much for your attention, and I'll take questions.
So thank you very much, Tom. Actually, you will not take questions yet.
Oh,
we,
sorry.
we will save the questions for the end, if that's okay with everyone. So please remember your questions that you have for Tom and feel free to ask them at the end of the last lecture here. So the next lecture is the research highlight, and that will be given by Manuela van Pinxerten. I always say your name wrong. Sorry. Van Pinxerten. This is correct, I think. Yes. Who's coming from Tropos, and she will talk about the sea surface microlayer. So the stage is yours.
Yes, hello everybody. Thanks for the introduction, and very happy to be here today and to tell you a little bit about the vital skin of the ocean, which is the sea surface microlayer. So we are all aware of the ocean and the atmosphere, but between these two compartments, there's still another very thin, almost invisible layer, and that is called the sea surface microlayer. And the sea surface microlayer, or short SML, is the uppermost 10 to hundreds of microlayer of the surface of the ocean. So why is that? Air and water have quite different properties. I showed here the chemical composition of air and of the oceanic water. And the compounds which are in the environment, they either like to be in the air or in the water, depending on their chemical composition. But some compounds have non-uniform properties, and they prefer to be at interfaces and actually form the SML of the ocean. And these compounds are called amphiphilic molecules or surfactants. And a surfactant has a hydrophobic region. This is shown here with this long non-polar tail, and a hydrophilic region. This is the polar head over there. And an example for a surfactant, which is also used very often in trace experiments, is nonanoic acid. And surfactants, such as nonanoic acid, they are in the interface, in the oceanic interface, like shown over here. So their polar head would stick into the water, and the non-polar tail would stick into the atmosphere. And with this, the surfactants indeed form the sea surface microlayer of the ocean. And this is also like the SML is defined. And this SML can more quantitatively be described via the enrichment factor, which can be calculated quite easily, as shown here. So the enrichment factor of a surfactant in the SML is defined via the concentration of the surfactant in the SML, via its concentration in the bulk water. And this is typically measured in a depth of 1 meter. An enrichment factor of 1 means that there's just a weak enrichment, or basically even no SML enrichment, because the concentration of the surfactant in the microlayer is the same like in the bulk water. An enrichment factor of 1.5 means that there's a moderate SML enrichment, and enrichment factor of 2 means that this is quite a strong SML enrichment, because then the concentration of the surfactant in the microlayer is double compared to the bulk water. Okay, how is the SML formed? So how are these surfactants brought to the surface? And the main process for this happens to wave breaking. So when waves break, then air bubbles are introduced into the water, as you can see here in this picture. And when these air bubbles slowly rise to the surface, then they can attach these surfactants at the surface of the bubbles. And actually not only the surfactants, but also other compounds which are associated to them. And then they are brought to the surface and form the SML. And this formation of the SML actually happens quite fast. And even when waves are breaking and disturb the SML, this upper transport of the bubbles brings the surfactants back. And so this is a process which really happens on a very short time scale, so within seconds. And this means that the sea surface microlayer exists even at high wind speeds and has therefore high coverage. So this is the main formation mechanism of the SML when it's windy. And when it's calm, then these surfactants get enriched in the upper layer due to diffusion, but this is much slower. The classical or the conceptual view of the SML is shown here. So it was originally thought that it consists of several layers. On the very top, you have this lipid or fatty acid rich layer, and underneath a layer which is composed of protein or polysaccharides. And underneath this, that there's the bacterioneustone, so that means bacteria which like to be at the surface, and underneath the phytoneustone. But more recent research has shown that the SML is probably more a heterogeneous matrix. So it's much more mixed and not such a layered, it has not such a layered structure. So, and it consists of these proteins and polysaccharides which form gels. And it's even described sometimes as a gelatinous matrix with microorganisms living right within. And this means that in comparison to the previous model, where it's a strict physical-chemical compartment, it's probably more a biogenic, rich compartment, and even described as a bioreactor. But independent of the model, it's important to know if the SML is relevant. So how is it globally distributed? Is it there all the time or just sometimes? And to answer this, there was a very important work which modeled the SML coverage. And this is shown here. The authors have used the enrichment of surfactants, the primary productivity, which is the main driver for the surfactant, so the biological producer, and the wind speed. And they have calculated in quite a simple model how the SML is covering the ocean. And the results are shown here in these pictures. So the orange part means that there is quite a strong SML. There's an enrichment above 2. The yellow part means that there is still a pronounced SML. The dark blue part means that there's a moderate SML enrichment, and in the light blue part, there is almost no SML enrichment. And we can also see that there's some seasonality there. And what is also obvious there from this model is that in the Southern Ocean, there's really almost no SML coverage. And this is due to the high winds there. However, it needs to be remembered that this is just a simple model to get a good idea of the SML, but there are much more complicated mechanism explaining and forming the SML. And recent studies shown that also in the Southern Ocean, there can be quite pronounced SML. So the SML can impact gas exchange between the ocean and the atmosphere. So you can easily imagine trace gases traveling between the ocean and the atmosphere. They are affected if there is a thick layer. And also chemical reactions happen inside the SML. There are some nice studies showing that when an SML is formed, that there can be, for example, a high formation of special volatile organic compounds, which really happens only in the SML. And also biological processes can be different in the SML. It is a layer which is exposed to a lot of harsh UV light, and the microorganisms they have to adapt. But the example that I would like to show you now is that the SML contains ice nucleating particles, INP. And an INP is a particle which acts at the nucleus for the formation of an ice crystal in the atmosphere. So an INP makes ice in atmospheric clouds. Okay, how does this work? So a cloud consists of water, which can be either there in liquid form or in ice form. And super clean water would only freeze at the temperature of -38 degrees in the atmosphere. But as soon as there's a particle, an impurity, the water freezes already at much higher temperatures, like, for example, -15 degrees. And such a particle is called ice nucleating particle, INP. An ice nucleating particle can, for example, look like this, such a long polyvinyl alcohol strain, which delivers a structured surface to the water. As soon as it touches it, it turns into ice. And what is known, that dust can be a good ice nucleating particle and forms ice at temperatures of about -20 degrees. and also biological particles are good INPs and form ice already at -5 degrees. And what was found in a very nice study is that the SML in the Arctic concentrates ice nucleating particles. This is shown here in this picture. I will only shortly explain what is done there. So to measure these ice nucleating particles, you take your sample and you freeze it down. So you cool it down and freeze it, and then you deserve at what temperature, what part of the sample is frozen, and then you obtain such curves. And what was found was that the Arctic SML really froze already at much higher temperatures compared to the Arctic bulk water and also compared to the SML in other regions. So this means the Arctic SML is enriched in ice nucleating particles. And additional studies also showed that these INP in the SML are very small and heat sensitive. So this means that this is probably some biological organic material. However, other chemical information of these INP in the Arctic SML is still lacking. So what is needed from a chemical point of view for a particle to be a good INP? You need to have many intermolecular hydrogen bonds at the proper position. So translated, this means you need a macromolecule which contains some functional groups like hydroxyl groups or amino groups in the secondary or tertiary structure. And this is shown, for example, here. This is a made-up molecule which would be a good INP. And looking at the classes of substances in the ocean, these characteristics transfer to proteins, carbohydrates, and lipoproteins, and also some others. And in our work, we wanted to study if we see a link between the carbohydrates and the INP. And for this, we have analyzed 3 monosaccharides like glucose shown there, and also these monosaccharides forming, which are bound together to polysaccharides. Yes, so these 2 kind of compounds we have studied. And for this, we went into the Arctic and sampled the SML and the bulk water. And the SML sampling is shown there in the other picture. We have inserted a glass plate into the water and removed it. And this is one typical technique for sampling the SML, because the SML is hydrophobic by nature, attaches to the surface of the glass, and can then be slowly taken up with a wiper. In the bulk water, we sampled with a self-made device of a telescopic rod in a depth of 1 meter. So this picture on the right shows the cruise track that we had. We went from Bremerhaven up north, and you can see that we had several stations there. We went into the pack ice, we were in the marginal ice zone, we also sampled the open ocean, and we also sampled melt ponds. And melt ponds, these are openings in the sea ice. So when the snow, which is on the sea ice, started to melt, it forms such lakes shown over there. And at a certain time, they break through to the ocean. And these melt ponds, they are very pronounced, especially in the Arctic summer. So we had all these many nice samples. We stored them at -20 degrees, and then we brought them back to our laboratories in Leipzig and defrosted them. And then these samples contained, next to our analytes, a lot of sea salt. And this is the problem for the analytics. And to this end, we performed a nice desalination technique via electrodialysis. And the concept I have shown there in this picture. So you put your sample in between membranes which have a charge, and then you turn on the charge, and then the ionized molecules, which is sea salt, like sodium and chloride, they travel through the membranes, and the non-charged molecules, which are our carbohydrates, they stay into the sample, and then we have a nice desalted sample. And then we measured in the sample the free sugars like glucose with a kind of ion chromatography. And also we performed acidic hydrolysis to break the polysaccharides into their single compounds, the single monosaccharides, and also measured them with ion chromatography. And here are the results that we have obtained. This graph shows the concentration of glucose in the different kind of samples that we have. So in the ice pack, in the marginal ice zone, ice-free ocean, in the melt ponds, and always compared the concentration in the bulk water and in the SMR. And what you can see is that the concentration are quite different within these compartments. For example, the green part in the picture shows the concentrations of glucose in the marginal ice zone. And we can see that the concentrations of glucose were quite high and also quite enriched. And in the ice-free ocean, the concentrations of glucose were lower and much less enriched. And we have done, we have measured the same compartments for the INP concentration, and the results look quite similar. And when we plotted the concentration of glucose with the concentration of the INP, so this is shown here the freezing temperature, this is the means for the INP concentration, we saw quite a nice correlation between these two parameters. And then we performed a cluster analysis. This means we sorted our samples based on their pigment concentration in the bulk water. And then we saw a really nice correlation between glucose and the INP concentration. And this is interesting because glucose itself is not an INP. It's just a simple sugar. And it does not contain these properties which is needed for an INP, as I showed earlier, like it's no macromolecule. So we were wondering, how is this connection between glucose and INP? What we know is that in the ocean, marine microorganisms are the only source for free glucose. So we thought of possible mechanism, how they could be connected to INP. And one could be that the algae cells, they actively emit glucose together with the INP. It could also be that the algae cells emit the polysaccharide and the INP, and then the polysaccharide are later then degraded. And that's why we see this correlation between glucose and the INP. And another idea is that glucose and the INP are inside the cell, and the freezing damages the cell, and the INP and glucose are released. And that's why we have this connection. So we don't know yet which mechanism is the real one, and we are still working on this. But what we can tell is that glucose and the INP are formed in the Arctic SML of the marginal ice zones and of melt ponds, and probably by microorganisms. And glucose is a good tracer for INP. So to sum up my talk, I have showed you that the SML links the ocean and the atmosphere, and all exchange processes between the ocean and the atmosphere, all trace gases, all particles have to pass this specific layer. But the SML is so far a concept. It's not yet fully understood and also not yet really included in ocean-atmosphere interactions. And I showed you in the second part that the Arctic SML concentrates biological INP and glucose, which is an INP tracer. But we also saw that Arctic Ocean is not the same as Arctic Ocean. These different compartments in the Arctic Ocean, so the melt ponds and the mids and the open ocean, they have quite different properties related to their INP and glucose concentration. And with this, I'm at the end of my talk, and I would like to thank you for your attention. And I'm happy to receive questions later.
Yes, thank you. Very good. Thank you, Manuela. Great. So now we're going to move on to the last talk in the session, and that is by Lisa Miller, who will talk about our cross-cutting theme. And she is coming to us from the Institute of Ocean Sciences, Fishery and Oceans in Canada. So Lisa, please.
Okay, thank you very much. Good morning, everyone from British Columbia. So now I am going to continue on the theme that Manuela started, talking about the polar oceans, which are not in many ways typical oceans. The polar oceans operate fundamentally differently from both tropical and temperate oceans. And the reason for this is because of the sea ice. The sea ice impacts all aspects of the polar oceans, the circulation, the ecology, and especially so air-sea interactions. Now, I know a lot of you, or at least several of you, already know a lot about this topic and are very deep into the subtleties and the details. And you may find yourselves a bit frustrated with how I skip over the top of this and oversimplify this subject. But the people I'm talking to today are those of you who have perhaps not really thought so much about sea ice and how it impacts the atmosphere and the ocean. So I have just two modest goals for this 20 minutes we have together. By the time I'm done, I'm hoping that everyone here will know two things. One, how sea ices form and how it's structured. And two, some of the ways in which sea ice can impact air-sea interactions. So to launch into this, other button, there we go. The formation of sea ice. Unlike fresh water that freezes from the surface, in seawater, it freezes within the mixed layer. Small crystals of ice form within the mixed layer and then float upwards. And this figure is a photograph taken in a tank in which seawater is freezing. And you can see the small ice crystals that have formed, that's called frazzle ice, floating up towards the top. But what's most important about this figure is you can see streams of dense water sinking below the ice crystals as they float upwards. And this is because the salts in the seawater are excluded from the ice. The ions simply don't fit into the crystal lattice, but rather they stay behind in the water, increasing the salinity and the density of the water, so it sinks downwards. And this is a fundamental process that we will very important process that we will come back to over and over again. So anyway, as all this frazzle ice is forming in the mixed layer and floating up to the top, it accumulates at the surface to form what's called grease ice. And this is what it looks like. It really looks like someone has smeared grease across the top of the ocean. Okay. And as more ice forms and floats up and collects at the top, it starts to consolidate into small flows. This is called pancake ice, and they're anywhere from a decimeter to a couple meters in diameter. And there's still a lot of open water in between. But as more ice forms and accumulates, eventually these small pancakes consolidate into larger flows that are thicker. Now at this point, the ice, you can walk on the ice. As a result, we have a lot more data from this kind of ice than any of the previous ones. We know a lot more about this. Eventually, even if the ice cover closes off completely with no direct connection between the atmosphere and the ocean, the ice still continues to grow slowly by water freezing directly onto the bottom of the ice. Okay, so it and that goes very slowly, but it continues through the whole winter into spring and even sometimes into early summer. Now, this process of ice formation, the sequence of events and the different kinds of ice leads to a very distinctive vertical structure to the ice, where at the top, where the frazzle ice has consolidated, the ice crystals, you can see at the very top of this polarized light image, the ice crystals are very small and oriented randomly. In the lower parts of the ice, where the ice is formed more slowly by direct freezing to the bottom of the ice crystals are much larger and oriented pretty much vertically. And then the other important feature about the ice is that sea ice is almost always covered with snow. It's very, very rare for there to be no snow on top of the ice. Now this snow is always salty. And this is again because of the salts that are excluded from the ice crystal as it forms. So the snow on top of the ice is salty, and it also often has a very rich biological community. And particularly if it has been flooded with seawater, either seawater flowing over the sides of the flow or even percolating up through the ice itself. Now, of course, this is a very simplified view. Often it's much more complicated, particularly if you're in an energetic part of the ocean, the marginal ice zone with a lot of waves. You can get multiple layers of frazzle and this columnar ice. But this is a good rule of thumb to think about how the ice is structured. So now, forward. There we go. Okay, so now I want to take a deeper look into what is actually in the ice. First of all, brines. Not all of the salt ends up in the underlying water or in the snow above. A lot of the salt is actually trapped within the ice as it forms in the form of concentrated brines that's trapped in brine channels and pockets. Now, I love this figure. It was produced at the Alfred Wegener Institute. They took a block of ice and impregnated it with epoxy and let it solidify, and then sublimed away the ice to leave behind a three-dimensional cast of the brine channel network. And you can see that the brine channels are mainly oriented vertically, but with a lot of branching in it. Now the structure and branching of the brine channel network is intimately dependent on the temperature. These are two experiments that were done. The left-hand side is light photography and the other is X-ray tomography. And they both show what happens as the ice is warmed from -15 degrees up to -2 or -3, depending on the experiment. And what you can see is at the low temperatures, the brine exists in very small pockets that are isolated from each other. But as the ice warms, those brine pockets expand and start to reconnect, so that as you're approaching the freezing point of seawater, the brine channel network is fully connected and the brines can move freely vertically through the ice column. In addition to the brines, we also have solid salts. This is the phase diagram for sea ice, with temperature along the x-axis, and the vertical axis is a logarithmic weight ratio scale showing the fractions of ice, brine, and solid salts within the ice at various temperatures. And what I want to emphasize here is at specific temperatures, different salt minerals form. First, at -2 degrees, we get a calcium carbonate mineral called icaite. At -8, mirabolite, a sodium sulfate mineral, forms. Another example is at -22, you start to get sodium chloride precipitating as halite. Now, at this point, the ion ratios in the remaining brines change dramatically. And so you can no longer consider these brines to be concentrated seawater. It really is a different kind of solution. Okay, in addition, we get gas bubbles precipitating out. As the temperature drops and the salinity increases, the salting out effect actually strips out the temperature effect. And the solubility of the gases decreases dramatically, and you get bubbles spontaneously forming within the brine channels. And that's shown in this, again, light photograph photos. I particularly love these photos because in the right-hand side, the small zoom ins, you can see at arrows 3 and 4, are pointing to mirabolite crystals that have also formed within these brine channels. And then the messy stuff, biology and organic matter. Organisms, cells, algae cells and bacteria also get trapped in the ice as it forms. The top photo here shows a diatom that was trapped in a brine pocket that continued to freeze and closed in on it and actually crushed and killed the cell. The lower figure, in contrast, shows a cell that's still intact and alive. The blue cloud around it is organic matter, and what's called exopolymeric substances, that's been stained blue to make it easier to see. And these exopolymeric substances are formed by both ice algae and bacteria. And they seem to act as both a osmoregulator and a cryoprotectant. Essentially, by releasing this material into the brine, it further lowers the freezing point and helps the brine pocket stay open, big enough for the cell to be able to survive the winter. There's also a very strong possibility that this EPS is from sea ice brines and organisms is a major component of those sea surface microlayer enrichments that Manuela was talking about. Okay, so we've seen that sea ice is an extremely geochemically and biologically active, rich and active medium. It actually plays a role in the biogeochemical cycles of almost all climatically active substances. So now I'm going to use the remaining time I have with you just to give you a few examples of some of the ways sea ice impacts these cycles. The first story I want to tell you is about halogens, about bromine, ozone, and a story of bromine, ozone, and mercury in the troposphere, the lower atmosphere. This top figure is a time series that was collected in the atmosphere at a station in the high Arctic. And what it shows is a year-long time series. The black dots are temperature. The blue is elemental mercury, which is a gas in the atmosphere, right? The red is ozone. And what you can see is that as spring advances, both mercury and ozone become extremely variable. And if you plot them against each other, which is the little inset, you can see that they co-vary. As mercury goes down, ozone goes down. As ozone goes up, mercury goes up. And what seems to be happening here is that in the spring, halides from sea salts, as they're hit with light, they create radicals which destroy ozone. This is the same reaction that happens in the upper stratosphere, but this is happening down in the lower atmosphere, in the troposphere. And this destruction of ozone produces halide oxides that then react with mercury to produce the elemental mercury, to produce mercury salts, which are not gases. And they fall out of the atmosphere, landing on the ice, on the water, and on the snow, including on land. Now you can imagine that this probably has some pretty significant implications for polar food webs, including the health of human communities that live around the Arctic. Now there's a big question here about whether these sea salt halides are coming directly from the water, from sea spray and bubbles bursting in leads and in the marginal ice zone, or if they're coming somehow from the sea ice. As I said earlier, the snow on top of the ice is really salty. And so that's a mix. But the question is, how would you get that salty snow into the air? And the answer to that is with winds, blowing snow. And this was a study done in the Southern Ocean on the edge of the Weddell Sea. showing, again, atmospheric measurements over a day and a half through a storm that came through. The top plot, wind speed is in black and temperature is in blue. The next two plots, B and C, just show particle numbers in the air, snow and aerosols, with the red dots being close to the surface and the black dots being 30 meters up. But what I want to focus on is plot D, which shows the composition of the aerosols. The bars are sodium concentration, but what I want you to focus on is the squares, which are the depletion factor of sulfate. So this is how much the sulfate is depleted relative to seawater. So above zero, they're depleted. And what we see is most of these aerosols are depleted in sulfate. If they had come directly from sea spray, they would not be depleted. The only way for that depletion is for these aerosols to have somehow been derived from sea ice in which mirabolite, that sodium sulfate mineral I mentioned earlier, has precipitated. Okay, you can see the next plot down, E, shows the composition of the snow. The black squares are the sulfate depletions. So, and so it's clear then that the source of these aerosols is the blowing snow from on top of the ice. Okay, now I'm coming down to the end of my time. So I want to spend a little bit time now also talking about air-sea fluxes, direct connection between the air and the ocean in the presence of the ice. The data on gas fluxes in the presence of ice are actually conflicting. First, lab experiments. This plot shows the gas transfer velocity on the y-axis versus fraction of open water. And the line shows what the gas transfer velocity would be if it scaled directly to open water. So 50% of the water is covered with ice, you'd expect the flux to be half. What this shows, though, is that the fluxes are actually all larger, quite substantially larger. Then out in the real world, again, gas transfer velocity versus fraction of open water. The red triangles are what you would expect based on the laboratory experiment. Again, the line is what you'd get if it scaled directly to fraction of open water, and the data fall right along that line. Okay, well, we've got the real world versus the lab. Yeah, that we can get pretty clear which one's probably right. Except that this is also the real world. This is measurements of radon deficits. Radon is a gas. Radon-222 is a gas. That's the radioactive decay daughter of radium-226, which is a salt that's dissolved in seawater. So when radon activity is lower than the radium activity, that shows there's been gas transfer, and you can calculate a gas transfer velocity. And so this plot shows, again, the gas transfer velocity versus the wind speed, but these have already been corrected for fraction of open water. The lines show open water parameterization. So if the gas transfer velocity directly scaled to open water fraction, they'd fall along those lines. What you see, some are really high, some are really low. There's clearly something here we don't understand, and this is a major source of this is a big field of study right now, trying to tease apart where this problem is. Okay, finally, just real quickly, I want to say something about ice water fluxes, the brine sinking out of ice. This is a really important factor in global circulation and deep water formation, and there's a lot of laboratory and circumstantial evidence saying that it's really important for carbon export as well. But this has been extremely difficult to measure. Right now, we only have one way to directly measure ice water fluxes, and that's underwater eddy covariance. The eddy covariance that Tom talked about, just you do it upside down underwater. So far, we only have the instrumentation to do this for oxygen, and the small amount of data we've gotten so far are promising. They make some sense, but this is definitely bleeding edge research. This is delicate instrumentation that does not respond well to either ice movement or interactions with wildlife. But another exciting field that we're exploring these days. So with that, I will wrap up by saying I want to emphasize that the sea ice is going to continue to be an important factor in the polar oceans for the foreseeable future. Although there are predictions that the Arctic will be ice-free in the summer within the next couple decades, there are absolutely no predictions that the ice is ever going to stop forming in the winter. We are going to continue to get a lot of sea ice formation going on. And so hopefully what I've tried to do today is just give you an idea of some of the challenges we're facing. And there are undeniably a lot of very interesting and also very useful things to study in these environments. So with that, I will stop and hand the floor back to Krista. Thank you very much.
Great, thanks, Lisa. So those were three very, very interesting talks. Thank you guys very much. And I hope that now we can open the floor to some questions from the students. If you have any questions, please raise your hand, your virtual hand, and I will call on you to unmute yourself and ask your own questions. So I see nothing. I will wait a second. So if no one wants to start, I will start. I have lots of questions written here. I would like to start go in the order that we received our talks. So Tom, I was wondering if you could talk to the students a little bit about how we determine whether or not we are caring about an airside K or a waterside K. So how do we know what our K, which K we're going for if we're not talking about a total K?
Yes, that's a good question. It relates to that point I made about the solubility. So if you look at the dimensionless solubility, so that's the water concentration over the air concentration, and then you implement it in that, I guess maybe if I could look at my slides, what was it? One of the slide 6, are we able to show that? Bless you, Lisa. Maybe not.
No, he's pulling it up, he says.
Oh, okay. So I didn't give him any warning.
I wanted to get to the fact that we're often talking about KW when we talk about the fluxes. And so a little bit about why we get to that point. Yeah.
Yes, it's good to re-emphasize this. Yeah, have I got control? No, maybe next, just click it one over so we get a whole couple of clicks. Oh, there we go. I got it. Perfect. So if you look at the red box, you can see that you have that solubility term, the divided by Ka. So if you're using a very insoluble gas, then that term, alpha over Ka, becomes pretty much unimportant in determining the total gas transfer velocity. So all of your resistance to transfer, or your 1 over Kw, determines 1 over the large K, which is the total transfer. And that's why if I go back, if we compare these, this is the full, if you like, thin film model where you have resistance of on both air side and water side. So that's how you ought to do the calculation. And you ought to be seeing a large K, big K, which is the total transfer velocity. But often for an insoluble gas, you'll see it like this, where the water side is the main control, and then it will be a small Kw. And so for something like CO2, that's fine. The resistance is all on the water side, and so it's appropriate. But you will see evidence in the literature where people have misunderstood this and applied this type of approach to a gas that is much more soluble. And in that situation, you ought to be including the air side resistance as well as the water side resistance.
Good. So I think when we make our calculations for something like CO2, as Tom is saying, then we want to use the KW. But if we talk about DMS, then we might want to start introducing the air side. And if we go all the way to some oxygenated kind of volatile compounds like acetone or methanol or something like that, then we really have to talk about Ka. So just, I don't know, wanted to re-emphasize these points. It's important to remember that the Ks are different.
And to give you a sort of ballpark indication of the importance, with CO2, the air side is less than 1% importance. DMS, it's on the order of 5%. And then acetone, I forget, but it's more like 50-50, or maybe even slightly weighted towards the air side, I think. Anyway.
Good. So thank you. And I guess I wanted to ask one more question to Tom, just to give advice to the students out there that might not want to go out and measure eddy covariance, but they might want to calculate fluxes like they did yesterday in the practical. What would be the advice to give these guys for their calculations? So what kind of steps do they have to care about in their calculation of the fluxes?
But undertaking a bulk flux calculation.
Yeah.
So I think the advice is firstly to think carefully about this, as we just discussed, the solubility of your gas, and thus the K that you might use. But also there are a range of different parameterizations out there, and some may be more or less appropriate for your gas of interest. So something like methane, you can probably use the dual tracer parameterizations fairly comfortably, because the solubility of methane is quite similar to that of the dual tracers. But if you're going out and you're trying to do calculate the flux of something like acetone, then you might want to consider a different parameterization that has been maybe derived from eddy covariance measurements, which might have measured, if not the same gas, a gas that is similar in terms of its solubility. And then the second thing to think about, which I didn't get into in a lot of detail, is this delta C. So the assumption in this model, if you look at the bulk seawater concentration, once you go below that diffusive sublayer, which is only tens to hundreds of microns, the assumption is that the water column is fully mixed in the surface, and that what you measure at 6 or 7 meters is representative of the point just below the diffusive sublayer. And there are instances where that's not the case. So that might be driven by things like ice melt, introducing stratification at the near surface, or it could be driven by diurnal warming, so the warming of the waters during the day that then gets broken down over the night, and then reforms again the next day. And these can drive potentially quite significant gradients in the delta C that then would have an impact on the correctness, if you like, of the flux that you calculate. So that's a couple of things to think about.
Good. Thanks. I think also I just want to add a couple points is that we might, when you consider your problem, you should think about your time and your space scales, right? So
yeah.
What kinds of wind products do you want to use? And some of these parameterizations are based on smaller scale studies, and some of them, like Tom showed, are based on global values. So think a little bit about your time and your space scales in your computation. Good. So I want to ask again if anyone has any questions before I move on in the row with my questions. No questions for these guys. I can't believe it.
I have
Everybody
a
has to
question.
be shy. Yeah. Go.
Unless any students want to ask something.
Yeah, I don't see any hands yet. So somebody else besides me is welcome to chat around.
So I have a question concerning the surface microlayer, which is a fascinating subject. So you defined, all your definitions are based on the surfactants and both or definitions and also formation mechanisms. Can't you define the microlayer based on, for example, enrichment factor of a material that are not surfactants? This is one question. And also, aren't there mechanism for microlayer formation that do not involve surfactants? For example, due to just buoyancy of very light material that accumulates at the surface. So I hope it's clear.
Yeah, sure. Very good points. So what is textbook knowledge, as far as you can say so, for a phenomenon like the sea surface microlayer? It's based on surfactants, and this is an easily understandable subject. And so it's mostly at first explained via these surfactants, because this is something you can think of as a surfactant likes to be in these interfaces, and it is there and has been measured there, and it's a good means to define the SML via the surfactants. But of course, and I think I shortly mentioned this, that also other components are in the microlayer, and there are several ways and several mechanisms how they end up in the microlayer. And one is, I think I slightly showed this, when the surfactants are transferred upwards with the bubble, with the air bubble, and form the microlayer, then a lot of other components can be attached either directly to the surface of the bubble, or also then attached to the surfactants. This is then called co-adsorption, and be transferred via this way in the microlayer. So not only surfactants are transferred there, but also other components. And actually, in these days, the microlayer enrichment is calculated for a lot of components, for polysaccharides, for DUC in general. So this is something that is done. And yes, you asked about the mechanism for the SML enrichment, and I also presented them, the two which are known. So one is this upwards transport via the air bubbles, and the other one is just diffusion. But what many also recent research is about, that within the microlayer there can also be specific for chemical or biological reactions which form additional compounds. So we have, for example, observed that some compounds which are not surfactant at all, quite water soluble, but they are also enriched in the microlayer. And an idea behind this is that they are really formed specifically inside the microlayer.
But they're necessarily or probably lighter than water, right? So the floating is or buoyancy is an important factor, right?
Yes, yes. Buoyancy is also something why these marine gel particles like TAP, they can be, when they are unbalanced, unattached, they are very light and they float on the microlayer. And this is another mechanism or another way how compounds can end up in the microlayer. Actually, the TAP are a very good example, because they can form the microlayer when they are light, but when they are attached to heavier particles, they sink down. So they can have a lot of walls within the ocean.
Thank you
I'm
very
going to
much.
interrupt. I'm sorry to interrupt. But we have lots of hands now. So I'd like to get the students' questions, if that's okay. And Yoav, keep your questions. So Anna, you were the first.
Yes. Do you hear me?
Yes.
Great. Thank you so much for all of your talks. Very interesting. And I have a question for, I guess it's both Manuela and Tom. Within these boundary models, is SML included? Could that enhance or hinder gas exchange, basically? Like, how is that accounted for? Is that, yeah. If you understand what I meant. You can decide who wants to talk. Thank you.
Maybe Tom, you want to start for the modeling?
Okay. So from a model point of view, I think that's part of this idea that you start from a very simple model that has this diffusive layer, but ultimately all of the processes that might influence the rate at which a gas transfers across that zone, or indeed is formed in that zone, will be encompassed in that K term. So there are gases, for example, ozone, where we think that the processes are just fundamentally different because the ozone is tending to deposit on the sea surface, but it's depositing in part at least because of chemical reactions occurring right in that layer. And so the deposition velocity, if you like, how fast that ozone is depositing is actually much more controlled by those reactions in the microlayer. And that's fine, that can still be wrapped up in that K term, but it's going to be very, very different when you look at something like ozone, which is reactive and is influenced by that diffusive layer or that microlayer, than it will be for other gases.
Yes, and if I can add to this maybe, so modeling and the SML is a very complicated topic because for doing this correctly, then the SML first of all has to be really understood. What it is, as I said, it's a model, it exists, but there's not so much knowledge like when it's there, what does it really contain? And to really bring it in a model, you have to see, okay, what are the important driving factors of this SML for several compounds? And what I can tell in terms of aerosol formation processes, I am aware of one model which includes the SML for the formation of aerosol particles, so from sea spray particles. This includes the SML as an enrichment compartment. So it is known that several compounds are enriched in this microlayer, so they have a higher concentration there compared to the bulk layer. And this is included in a kind of a sea-air interaction model, or let's say in a parametrization scheme. So this is one I know which is currently developed. But other than this, I think it's really complicated and there's still a long way to go. As I said, it really has to be understood like what really is the SML under these diverse conditions.
Okay, thank you. The next question is from Brandy.
Yeah, hi. Can you hear me? Okay, good. So I have to ask questions about the SML in the Arctic because my poster's on this, and then you both Lisa and Manuela talked about it. So I'm curious because, I mean, I guess I'll do it in my 1-minute presentation tomorrow, but I really expected to find a very enriched SML when we went out there, and I didn't. I had sort of low concentrations of my EPS particles, which is why I study. And in my work of trying to figure this out, I've kind of really started to understand how different an environment an open Arctic open lead is compared to what we understand for the SML on open oceans. So I'm curious if you, like, particularly too, if you can just kind of talk about that, like about how the different processes are going to affect the SML in that kind of environment, whether it's, you know, ice leaching out, meaning that you're going to have more near surface kind of production occurring that's going this way, or if you're going to have more of these sort of wind driven forces and physical mixing that will occur there.
Yeah.
Okay. Maybe I can start.
Yeah, go ahead, Manuela.
Okay. So, hi, Brandy. First of all, great to see you and looking forward to your poster. You said it's tomorrow, right? Okay, perfect. Yes, SML and the Arctic. So this is really a very exciting topic. And this is also something that I found really amazing when we discovered this, that so you can measure you can probe the SML in the Arctic in several regions and find different results. And we were surprised to see that in the open ocean, in the open Arctic Ocean, that there was quite a low enrichment and concentration of these carbohydrates that we measured, which are similar to the TAP and AP compounds that you are measuring. And they were really high concentrated in the marginal ice zone and in the melt ponds. So for this reason, we think it's in a way connected to freezing and deep freezing processes. So I showed a slide where we thought of some mechanism which could be behind it. And I think it's really, it's about biology. So there are biology processes. And actually I have a question about this for Lisa later, if I may. And that within the sea ice, these compounds are formed probably due to this high biology in the sea ice. And when it's then melted and frozen again, what happens in the midst and on the melt ponds, because they are exposed to a lot of these changing processes, that this really releases these compounds. So it's a mixture between a biology and a physical process. So that's what we think from the data we have. And this is not happening just in the open Arctic Ocean. So it needs to have the sea ice combined with productivity and melting. So that's what I can say to this. And I'm sure Lisa can add something there.
Yeah, and I'm just thinking out loud here. Another possibility is that if you're in an area of active ice formation, like in a lead or a polynya, you could actually have the new ice, the frazzle ice, hoovering up the surface as it's blown across and consolidated. Hang on a second. Sorry about that. It's breakfast time here. So, and the other important thing to think about the Arctic is that you've got river waters, which have a big impact, and the impact from the rivers can actually extend a long way under the ice and emerge far from the river mouths. And the geochemistry of the river waters and how they impact the microlayer is completely different from what's in the sea ice. And we've done a number of we've had a number of expeditions where we found low salinity and found high INP concentrations in the microlayer, and we thought it was ice melt. And then we went back the next year and actually got our oxygen 18 measurements right and discovered that no, it wasn't sea ice melt, it was river water with the high INPs. And so, yeah, not simple. Not enough of us measuring it either. So good you're working on it, Brandy.
Yeah, actually, I think that's the title of my poster, is like novel measurements, because we were able to take the catamaran up there and get large volumes. So we took like the first chlorophyll and particular organic matter stuff, and that was the cool thing, was getting that for the first time. But yeah, this was just an interesting thing, is because I think we had our surfactants stayed the same, but then our gel particles went down. And so it just created this question to me of like, okay, what are the actual drivers in an Arctic environment? Because I'm not used to this. I'm used to open ocean, and I know how things work there. And so I was just curious when you both start talking about SMLs in the Arctic. And I also learned, this is kind of a question for you, Manuela, because I also took my underlying water from 1 meter. And then later on, it occurred to me that the ice edge goes down to about 3 meters. So I realized what I was actually sampling as my underlying water is a distinct sublayer, is what we've called it. We're like, this is a distinct sublayer between the underlying water. But I'm curious if you guys did anything else, because we basically just had some ADV measurements below the ice where we could kind of show what the currents were doing in the area. But did you guys have any measurements, any kind of current measurements or physical oceanography at deeper depths to know, I guess for your open ocean one, this is fine, but the closer you get to the ice, the more this is going to be impacted, right?
Yes, this is another very complicated topic, not only the SML, but also the bulk water. So I said before, what's typically done is taking the bulk water in a depth of 1 meter as a reference value. But this has to be possible in the Arctic, because when you have a melt front, and sometimes it's even not 1 meter deep. And then you also would assume that this bulk water is very well mixed, which is not always the case, as Brandy knows very well, because she has studied this. So yeah, this just adds more complicity to the problem. And I must say, so for our measurements, we really just took the sample in a depth of 1 meter to be comparable also to other systems. But this is certainly something where you could look into in more detail. So it's not getting easier, it's getting more complicated.
Okay, thanks. I'm done.
Great, thank you. Okay. Wan Shu, do I say your name right? Right? Sorry about that.
Yes, thank you, Kristen. I have a quick question for Lisa. So Lisa, you mentioned use the eddy covariance for the water side gas flux measurements. this is pretty cool because we generally use the eddy covariance for the water side gas flux measurements. But I'm wondering how can we do that? Because for the eddy covariance, it requires very high frequency measurements, right? So can we do the gas concentration measurements in the water side, like 10 Hertz, something like that?
Well, the thing is, one of the nice things about water side eddy covariance is it doesn't need to be as high frequency because the eddies in the water are moving more slowly. However, oxygen's the only one with high enough frequency yet. We can't do CO2 because the CO2 sensors in water aren't high enough frequency. So yeah, you have to match the frequency to the medium. Now, if we wanted to do gas exchange in molasses, we'd be set.
Okay, makes sense. Thank you.
Please don't do that.
All kinds of images of molasses ruining instruments. Can I ask a little follow-up on that? Because this is a nice question from Reggie. Do you know the frequency that needs to be measured?
No, I don't. I don't do that. Even though that was my name on the caption, that was a synthesis paper that I was the lead author on. Brent Else does those measurements, and yeah, I don't have that frequency at my fingertips. I'm sorry.
Sorry. Thanks.
Okay, thanks. So Margaret? Can we have Margaret?
Yeah, hello, thank you very much.
Yeah, there she is.
Yeah, thank you for the opportunity. Yeah, I just want to ask a question. It's based on also my poster, which I'm going to present later, sometime now today. Yeah, it's the winter carbonate properties in the Southern Ocean. That's the South Atlantic towards the Southern Ocean. Now, I want to ask, we have heard from the first speaker talk about the solubility constant, depending on temperature, and the flux also depending on the K and the delta C, that is the concentration. Now, I'm wondering, we had this sampling, we had it like 2 weeks in between, the same region, the subtropical region. At the first sampling, we have like maybe sources of fluxes into the atmosphere, more of carbon dioxide source into the atmosphere. But on the return, in 2 weeks' time, we are having more of carbon sink into the ocean, but we are having the same temperature. And even the DIC, there is no much difference. So though we didn't look into the biology, but I don't think there was much difference in the biology going just by sight, but not by science. You understand what I mean? So, but I'm still thinking, what could be the reason or the driver of the process that you are having a carbon sink between 2 weeks and you have almost the same temperature range? Yeah.
Yeah. So did you say the DIC was the same as well?
Yeah, almost the same also. The DIC was almost the same.
Okay, well, that implies that the alkalinity must have been different somehow.
Okay.
Yeah, now see, if we were in the Arctic, I would
go in
have
the
said,
Southern Ocean?
Yeah,
Okay.
yeah. If we were in the Arctic, I would have said you've moved from an area that was dominated by sea ice melt to an area that was dominated by river water, where the geochemistry is completely different and the alkalinity to DIC ratio is really different.
Okay.
It's seawater versus
Okay.
river waters. The Southern Ocean, I'm not as familiar with it
Okay.
where you were, but that is something I would look for. Look for a different water mass. Look at the alkalinity to DIC ratio,
All right.
and could a front have come through that area? That
Okay.
so that would be the first thing I would look for.
look at. All right.
Maybe
I
one of
was
the others,
actually
Tom, I see Tom has unmuted, so he might have a brilliant idea.
Well, I don't know if
okay.
it'd be
Thank you
brilliant.
so much.
Well, just you mentioned about the direction of the flux, and of course that I wondered whether is that with exactly the same atmospheric concentration, or are you using the whatever you happen to measure? And could that you can get fluctuations? I mean, I know it's the Southern Ocean, but you can get fluctuations that might have driven it.
Yeah, the atmospheric concentration, we use the average.
Okay,
same
so
thing.
it's not driven.
Yes,
Okay.
the same
And then
thing.
my other suggestion is, of course, rainfall could be the other impact. If it rained in between, then that might introduce a surface lens that then very quickly gets mixed down, but that could have an impact.
And chemically, rainfall looks a little bit more like river water too. It's a lower pH. Yeah, that's
All
a
right.
good thought.
Right. All right. Thank you so much. Yeah. Okay. I think I got some idea. Thank you. Yeah.
Okay, Lucas.
Yeah, so I have a question for Manuela. So you mentioned, or you said that the surfactants have an influence on gas exchange. And I was wondering what the main processes, or what I thought to be the main processes that influence those gas exchange. So are those biological or physical processes, and also in which direction they influence the gas exchange? So is this a negative or a positive feedback?
Yeah, very good question. I know there are a lot of studies about this, especially how CO2, how the flux of CO2 is influenced by an SML. And I am aware of some studies who have really measured surfactants in a tank and in the ocean, and so different surfactants concentrations, and then looked at the effect of CO2. And so it's a physical impact from this side, because you just have a thicker layer and it will slow down the diffusion from both sides. So either if the gas goes into the ocean or goes out. And so from one study, I know it was the outcome that it really reduces the uptake. So the surfactants reduce the uptake of CO2 from the ocean. I don't remember the numbers, but it was a certain amount. And yeah, so that's what I can say to this, but maybe Tom has some additions there. He unmuted.
Yeah, just to add that, so generally gas transfer has been parameterized with wind speed, but what's actually happening is the wind is introducing, or there's shear at the surface, and it's introducing some turbulence in that diffusive layer that helps to refresh that layer and reduce the resistance to transfer. Surfactants can modify the viscoelastic properties of that layer so that when the wind blows on it, that transfer of turbulence is kind of dampened. So what that actually means is that for a situation like that, the direction of the flux is not necessarily fundamental, that the gas transfer itself would be modified whether it's going in or out. But you can have other situations where the surfactant properties themselves might chemically modify what's happening, and that might then have more influence on an influx versus an efflux.
Great, thanks.
Yeah, all these questions are great.
So, Samira.
Hello. Hello, so thank you for the presentations. It was really interesting. So my question is for Manuela. I'm wondering, I would like to know if there have a different method of sampling procedure, and if this method can affect the calculation.
Very good question. The SML sampling, yeah, it's it's a topic you can make an own lecture about this. There are several methods for the SML sampling, and I shortly showed this glass plate sampling, which is a very typical one. It's quite easy to apply. It's one of the advantages, and so you can easily bring it to the field, and it works very well, and it's good characterized. I showed before, you dip a glass plate into the water, and then you slowly remove it at a certain speed, and then the hydrophobic SML is attached to the glass plate of the wall and can then be taken off. And it has the advantage that from the, so you know the area of the glass plate, and you can see, or you see the amount of SML that you have sampled. And from this, you can then calculate the thickness of the SML that you have sampled. So you get a bit of a quantitative parameter. So this glass plate technique is really very often used, and you can do this either manually, which where you get strong arms, or you can also apply this on a catamaran. So you have the catamaran, and it's these glass plates are attached there, and it's an automatic sampling device then. So it's the same principle, but you have this, yeah, it's more elegant to work with this. Aside from this, there are other techniques, and I'd like to mention one other, which is the Garrett screen. So there you have, it's a net of Teflon, which openings, and then you bring it into the water surface and slowly remove it, and then the SML is sampled within the openings of these Teflon net, and then you can just easily, yeah, take out the liquid. This is another technique which is used, and this has the advantage that you don't have to be so close to the water surface for sampling. So, but you asked for the standardization, and this is a very good point. The glass plate is somehow a bit standardized. For the other techniques, I'm not so aware of, but this is really also a big need which was found in the SML community, that there has to be yeah, more really procedures how to handle it for having a better comparability. So it's, for example, known which volumes you can sample with the glass plate and which volume you can sample with the Garrett screen. So this has to be documented. And if alone from this, if you have a difference in the sampled volume, then it's already difficult to compare your results. And the SML community is aware of this, and there has been a very nice sampling guide published some years ago already. You can find it in the internet. So it's a sampling guide for SML techniques, which describe. Sorry?
Is it Whirl? Is it Oliver?
Yes, it's Oliver Wall, and I think with Michael Cunliffe. So I can write the names in the chat, so you can find. I think it's yes, exactly. Thank you, thank you, Krista. So they have published a really nice sampling guide which describes these different techniques and the advantages and the disadvantages, and point out that there really has to be more standardization for this.
Okay, thank you.
You're welcome.
Great. Oh, we have another question from Lucas.
Yeah, I have a question for Manuela again. So you mentioned that acid at the very beginning of your presentation. That is often used in studies as a surfactant. And later on, you mentioned that the surface microlayer is indeed a very complex structure made of many different molecules. So I was wondering how you can make sure that this acid that is often used has all of the, or is a good proxy for all of the different chemicals that are in the surface microlayer?
Yeah, well, it isn't. So that's just a point. So I wanted to underline that non-anoic acid is a surfactant which is often used in studies for surfactants in general. That's what I wanted to say. And you made a good point because this is how to study the SML in the lab. And this is not trivial. And what is done sometimes is that you take surfactants like non-anoic acid, add it to your water, and then assume you have an SML. There's some noise. Okay. And then do your experiments. But this is a bit difficult because it has been it is known that the SML is much more complex than this. It's not just a surfactant being enriched at the SML. This is how the first model was about. It's more this really gelatinous layer, which is, has these gel particles, has biology inside. So it's really, really difficult to produce something like this in the lab for dedicated lab studies. So thanks really for asking this, because I really didn't want to say that non-anoic acid is a good proxy for the SML, but it's a proxy used as surfactant to study impacts of surfactants. But studying the SML is really, it's much more complex issue which has to be accounted for.
So is it then because it's chemically similar to the surfactants, or why do you choose exactly that acid?
Non-anoic acid was, I choose it just an example because it's used in many studies that it's a good surfactant. So it's a good available surfactant, and it also occurs naturally, which is used for many kind of experiments. Tom, maybe you can add.
Well, just to add that it's a common problem. So when a lot of gas transfer work has also used Triton X as and again, it's a reference surfactant, and so that has been very useful for understanding what the impacts might be and the magnitude of those impacts. But fundamentally, how well does it represent what's going on in the ocean? It is much more difficult to, and then that of course could change depending on the organic composition of that microlayer. So generally it's a reference. It enables you to compare with different studies, enables you to get a sense of how important the process that you're looking at might be, but not a fundamental right answer.
Okay, thank you both for your answers.
Maybe Lucas knows this already, but you gotta get these guys like Bantina to put seawater in their tanks, and then you have less of a problem, right? But this is hard. It's hard. Okay, so Santiago had a question. I think there's time for one question.
Oh yeah, sure.
No,
I mean,
sorry. I
don't
just saw your.
Okay.
Thing, sorry.
That's
we
okay.
don't. We can go to break. That's fine. Okay, good. So we're basically anyway at the time to go to break. Santiago is busy in the next 2 hours, so I understand if he wants to take a break. So thank you guys. Thanks to the lecturers for this session. It was really great, very interesting. And thanks to the students for asking a bunch of questions. Yay, that was also really great. And we will come back at, I think, the top of the hour for the practicals. So everyone is free to turn off their cameras and their microphones and take a 10-minute break. So see you soon.
Hello there.
Hi.
Hi. So is everybody ready?
Yeah.
Me and Santiago.
That's what I mean. Everybody.
I don't.
Okay, so I guess Paul will be back soon. Or maybe he's
he
said already that he's ready. So there are 39 people here. I don't know if we should wait a minute. It's not technically the top of the hour yet.
Do you hear background noise from my end?
No.
Okay, because I have family all the way there talking.
So no, I don't hear it at all.
Okay, good.
I'd like to wait a minute to see if more people come. I don't really see more people coming. Okay, but now it's technically the top of the hour. Well, let's just get started and we see if anybody jumps in. So the idea now is that we switch over to the practicals, and that they will be led by Santiago and Yoav. They're here ready. And I believe, so guys, correct me if I'm wrong, but I believe you will give some introductions and then to the plenary, and then the students will be broken up into breakout groups, and you will jump around to the different breakout groups to help them. This is my understanding. Yeah.
Right.
Okay,
Yeah.
Then I give it to you and enjoy.
Thank you. Okay, again, I'm Santiago, and this is Yoav, my partner in crime today. We're gonna be talking about satellites. This is a very short window for talking about satellites. So we are just going to give you a sample of what you can do, particularly using internet portals and how to look at satellite pictures. And we are going to do some practicals related to that. So we both prepared a couple of brief introductions. I'm going to be talking more about satellites in general, just give you an overview idea, and then Yoav is going to give you more specifics of the practical we're going to be doing. So I'm going to switch to presentation mode. Let me see, share screen. Here. Okay, let me this. Okay, is it sharing? It should be. And now let me go presentation. There it goes. Okay. So I'm assuming you guys can see this. So I'm going to be talking about, just to give you some perspective, at least all my years of working with satellite images and things you should keep in mind when working with satellite images. So essentially, one thing to remember is that when we are working with satellite images, we should remember not to be so biased by the how pretty these images are. And it's true. I mean, looking at with those images from space, some things can be very attractive. I mean, objectively attractive. But when we do science, I mean, there are more details that we need to keep in mind. And one is what is going on behind those images, not only in the physics and geophysics that we are working on, but also be very mindful of what we how those images are generated. And so that's one aspect. And the other aspect is we have to keep in mind what we want to ask. I mean, we have this fabulous image, what we want to address with that besides, you know, just the purely description of what we are seeing. And this is some of the perspectives I want to give you when looking at images and satellite images. So for example, one thing about satellite sensors is that in a very simple way, we can think about technology we have already in our cell cameras, because essentially satellite images, particularly those that just take pictures, are just like very sophisticated cameras like the ones we have in the phones. For example, they're very fast. I mean, remember, satellites are flying about 400 to 800 kilometers above the surface. They're going very fast, which means you have to take a picture very fast. So they're really sophisticated instruments. You know, they're taking, you know, tens of pictures within a second. And because they're going fast, yeah, that determines your spatial resolution, for example. You have lots of pixels. I mean, that's also determined by the spatial resolution and the speed of the satellite, that relates also what we know already from phones. And then, but this is another aspect, is these cameras degrade with time, and that's where the issue of calibration is so relevant, particularly those who are looking at geophysical effects that change in time, you know, temperatures and concentrations of any parameter you're looking into. You want to know that what you measured this year is equivalent to what it was measured 5 years ago, and that depends on the calibration of the instrument. Is critical, and that's actually what determines the quite a bit of the budget of designing a sensor. It's just making sure that the calibration will be consistent through the lifetime of the instrument. Is what really drives the cost of the instrument when you send it up. So these are all important considerations that you know, when we're talking about satellites, are happening, and what helped to create that pretty image that you have at your hands. So there are a lot of engineering happening to get that to your hands. And just a couple of very basic stuff. Remember, there are essentially two technologies to do satellite remote sensing. One is what is called the active remote sensing, which is the most common analog will be a space radar, where a signal is sent out from the sensor, and you measure the return. So what is essential here is that you know what you're sending out and you know what you're receiving. So it's from that change is that you make inferences about your object, you know, clouds, surface, whatever you're looking at. So that's called active remote sensing. And then you have the more, maybe more, we are more familiar with, is called the passive remote sensing, where the source of radiation is coming from the sun. And we already have independent measurements. So we know how much the sun is sending out towards the surface, to the earth. And, you know, our camera measures just what is coming back, what we call the radiance. And again, whatever is reflected or whatever is not coming to the sensor is where we make the inference of what is out there. So just two very basic things. One is about two types of satellites you can find. One is the polar satellite, which is these polar satellites, they fly, or what, they don't fly there because there is no atmosphere, but they're orbiting at between 400 to 800 kilometers. What the main feature is that because they're flying, orbiting so lower, you can have pretty good spatial resolution from 1 meter, like the Planet Lab satellites, or to 500 meters, like MODIS, things like that. But then also because the design of the orbit is such that you only pass over a given place once or twice a day. And this contrasts with the geostationary satellites, where they're orbiting much farther, 30,000 kilometers away from the Earth. The spatial resolution is limited. I mean, having 1 kilometer resolution at geostationary is quite stretching what the technology allows you to do right now. But also what you can do is you can take pictures every few minutes. And that's what is driving the design of these geostationary sensors. They're mostly used for weather monitoring, you know, hurricanes and rapid storm. And you can use these sensors with 1 minute sampling to 10-minute samplings, and you can see how the storm evolves. So this is just a big picture, nothing very, but it's give you an idea of what sort of different sensors you have available and when things go into place. The one thing is that about satellite measurements is that the product that you get is not necessarily is a direct measurement. Most of the satellite observations are indirect. With this, I mean, basically what you observe is, let me see, do you see my mouth, the pointer here? Yes. You have this is what you measure, the top of the atmosphere radiation or signal that you get, which is made of two components, atmospheric or ocean surface. And according to your discipline, you're interested in one of them. In the case of the atmosphere, if you're interested in ocean surface, you have to somehow get rid of the atmospheric contribution, or vice versa. So in the case of the atmosphere, which is what I'm interested in, this component is actually a function of several properties of what I'm looking for: aerosol concentration, which I want to find, optical properties that in principle I do not know, the geometry, the angles I'm looking at. So these are the way we deal with this, and the way we generate the data that you eventually use is by using models. And basically, if you are an oceanographer, you're gonna do a modeling of the atmosphere and subtract your model atmospheric component from the total observation of the radiance, and you get your ocean surface radiance, and from there you make an inference about ocean biology, surface temperatures, etc. Vice versa with if you want to do something regarding the atmospheric measurements. So for example, here we have a nice image from a dust event off the coast of North Africa, and here we have a clear sky a little bit, and here I think this is a smoke actually. So, but this is a very common scenario you have in the Mediterranean. So one question you want to ask is, something that is very common we can do from satellite. What is the chlorophyll concentration? What is the aerosol concentration in those dust and smoke clouds? So basically what we do is first, actually, in order to have a derivation from this is we need to find those pixels that do not have clouds. So whatever we do, we have to apply some algorithm scheme. Could be artificial intelligence, could be some physical approach. And then what we have is here, for example, it's an example of what we can get. We can get chlorophylls, or we can get aerosol concentrations to the right. On the left-hand side, you have chlorophylls at the surface. What it was done here is the observed radiance from the satellite there was a modeling of the atmospheric component, and from subtracting the atmospheric component, the chlorophyll was retrieved. And on the right-hand side is what we atmospheric scientists do. Is we do the opposite. We take the observed radiance from the satellite, we subtract an assumed ocean component, and then we get the whatever is in the atmosphere. So this is kind of shows the approaches that each community does. I guess the mess happens when you want to look at things in combination, in between, and get more precise at quantitatively. Also, for example, in these two images, you can see different patterns. Like, for example, on the left-hand side, you have here like this band structure. This is, for example, that it's a sign that the sensor is degrading. And also you can see more details in this ocean image, whereas here in the aerosol image, you don't see that many details. And that's two reasons. One is because the atmospheric researchers, or the algorithm group that created this aerosol product, aggregates the native pixels in a very different way. So it reports the data at 10 by 10 kilometers, whereas the ocean groups on the left-hand side report the data at 1 by 1 kilometer. So more details and more deficiencies on the instrument become more apparent. So this is all information that you need to have at hand when you're trying to make these inferences of data from these images. So just wrap up, what we're going to be using is just a couple of web portals. We're going to be using Worldview, and there you have the image, the web address. Worldview is one of the main portals available from NASA. Basically, it starts like this, showing you a daily global image of whatever is happening on the world. It's just this is called RGB, which is a combination of 3 channels, each visible wavelength. And then on the left-hand side, you will have different options to add layers of information, like the concentrations that I showed earlier. And then you are going to use another NASA portal called Giovanni. This is it's a little bit different in the sense that you have more freedom to select the data and display it. And in this way is more data oriented, but at the same time, it doesn't have the nice displaying options like you have in Worldview of adding RGB images. So we're going to be giving you examples of how to play with both and pay attention to different ways of interpret the data, because there are instances where it's just you may be misinterpreting because you don't have enough information about the sensor, or just actually none of the portals are perfect. So there are a few things that you need to know in order to make a proper geophysical inference. So I think that's it. I'm gonna pass the torch to Joab here. So I'm gonna stop sharing.
Yes, now it's my turn to share, right? So let me do it. Thanks, Santiago. It was very good, very nice presentation, and no, but very useful. It's exactly this setting the stage to what I will say. So hi everybody, I'm Yoav. As Santiago said, I work in the University of Haifa, and Well, and I come from the oceanic side, so we are rivals. My signal is Santiago's noise, and Santiago's signal is my noise in terms of remote sensing, as Santiago just mentioned. And so, in this practical, we have these are the overarching goals of this practical. First, we would like you to get familiarized and to feel comfortable, start feeling comfortable using remote sensing data. Of course, for people who are experts in remote sensing, this wouldn't be much of a use, but to our understanding, remote sensing can be used by these days by almost anyone. Much of the data is freely available, is highly accessible, and we think it can be used by at least as a reference or for providing geographical context in almost any research. We would also like you to practice a little bit the characterization of spatial and temporal variability patterns in satellite data in various SOLAS variables. So as Santiago said, from satellites we can have both atmospheric variables as aerosol optical depths or cloud properties, and marine properties as surface chlorophyll concentration and sea surface temperature. So we would like you to get some little experience in using these, in analyzing or at least observing this variability pattern. And finally, we would like to explore the nature of or the possible explanation to covariability between atmospheric and oceanic variables as observed, or oceanic or atmospheric observations, satellite observations. And as Santiago said, we can think of three possible explanations for covariability between ocean and atmosphere satellite observation. One is cause and effect. So one signal, for example, the ocean variable affects the atmospheric variable, and we have actually causality. The second is that both signals are driven by the same external forces, for example, seasonality. So we often have seasonality in both, similar seasonality in both atmospheric and oceanic variables, and this would result in covariability. And the third, and probably the most disappointing explanation, is retrieval bias. So as Santiago said, the ocean is the noise of the atmosphere, and the atmosphere is the noise of the ocean. So if you want to explain covariability, you need to overcome or to understand the possible of biases. We are now here. So as Santiago said, we will use these two data portals. I think you have them on your chat, right, Paul? Am I correct? So please go into these two, first open these two links. Note that for the Giovanni portal, you need to register. It is a very fast process, but you can do it also a bit later during the practical. But these are the two websites we will be using. If you know other portals or other sources that you're familiar with and you feel comfortable with, feel free to use them. So what is the assignment? The idea is to identify distinct spatial and or temporal variability pattern ideally in at least two SOLAS variables. So ideally one in the ocean and one in the atmosphere. And so this is the first goal, and the second goal explains the nature of the association. Again, I'm confused. Yeah. So here I have one a possible example. So these are satellite images of three types of satellite images. On the left, we have surface chlorophyll. On the middle, we have sea surface temperature, and on the right, we have, oh, it's the wrong title, sorry, it's the true color images, and here we see the clouds. Okay, now if we, something is wrong here. Okay, in writing the cloud, if we zoom in on the equatorial region, so the area marked by this red box, we can see that we have a distinct signature, both in all three variables. In chlorophyll, we have this high, very high concentration associated with upwelling in this region. In the equator. In the temperature, we have the opposite signature, which is due to this upwelling. And as you can see, in the clouds, we also have a signature. Here, the signature is a bit strange. The signature is an area of no clouds. You see here, right in the right-hand panel. So here we see a covariability, spatial covariability, and the reasons for it, it's for you to explain. Another fast example here is here of Peru. We have these decks of stratocumulus clouds. And if we look at the same time on the surface temperature, we see a distinct signature of cold temperature, cold water. Again, is it cause and effect, or is it a bias? We can discuss later. But so this is, until now I talked about spatial covariability. Then using Giovanni, you can also explore temporal covariability. And here we see a 2-year signal of different variables. So again, sea surface temperature on the right, sea surface chlorophyll on the right. You see that there is similarity between the signals. And also cloud fraction. It also has some similar seasonality. Again, what is the reason for this? The idea in this exercise is just to have an educated guess. Okay, so ideally you will identify such cases when you have spatial and or temporal covariability and provide an explanation. How do you do it? First of all, in both these portals, you can once you have a plot, you can create an image and download it. So make a presentation, a very coarse presentation. Just put the images on a presentation. So this is a deliverable. Okay, a presentation with nice images and educated explanation. So now let's go rapidly to worldview. I will shortly show you how to use it, and then I encourage you to, you will go into the groups, you will start exploring. First, identify interesting features from what you know of, from your prior knowledge, or just by looking for these nice features. And then we will visit you in the rooms, and we will try to explore and to guide you on or together to find to complete this assignment. So now I'm going. So whoever can, please go into the link, the worldview link, and I am also going there. No. Okay. So this is how. So when you open the worldview, this is what you will get. Well, you will get an image with nothing on it. Okay, just a RGB image or some image of the world. And then you can add layers. The way by which you add layer, you have this button here on the left-hand side. On the bottom, and you add layer, and you can type any keywords. For example, I want let's say chlorophyll. Oh, it's in Hebrew. And then I tag whichever variable I want, and then I add it here. And here, by pressing the I here, I can either put the variable or show it or make it disappear. This is one thing. The other thing is here on the bottom, you have the timeline. So you can choose which day over the period you want to use. So this is it. For example, here I have an arbitrary day of chlorophyll image, and I can go here to the equatorial and have this nice zoom in and have this nice feature here and start exploring it and see if I can understand how it is associated to other variables. I hope it's clear. And let's see if anyone has a question before we go into the rooms. So at the first stage, please just open these portals, try to look for nice, interesting spatial features, and try to make sense out of them. And me and Santiago will go through the rooms and start guiding you with the assignment. Yes.
Can I jump in? So there was the question in the chat. I don't know if it's important to discuss it now or if you want to discuss it later.
Okay, which one?
And Santiago said he could say something to it about the polar regions.
Again?
So there's another. Sorry. So somebody asked in the chat about the polar regions, and Santiago mentioned that he could say something to it.
Okay, go ahead.
You're muted.
Yeah, if when you in share my worldview part, but worldview you have a polar mode. If you go to the upper right, there is a little icon that shows a world. If you double click on the icon, you can see an option of projections here. Switch projection. You can see Arctic, Antarctic. So for example, yeah. So for example, right now, I mean, this week, for example, I was just goofing around, and there are big fires right now in Alaska, and probably observing Alaska is better to polar mode a little bit, just, rotate this way, and it will show it in more better way. All this mode, and you can put if you want. That's the fire. So here in Vancouver, so this is what you can do. I think, but the only limitation is that not regarding the polar regions is that worldview does not have all products in polar projection. So maybe sea surface temperatures that you have it in global mode projection, when you switch to polar mode, not necessarily all the data is going to be available. And that has to do with how the native data was delivered to worldview. Some native data was not delivered with the polar projection, so the worldview interface cannot do that. That was the comment.
Okay, I think we can go to the rooms and start playing.
Yeah,
and
let me get out of here.
I hope it's
stop sharing. Okay.
Okay. So so we split into rooms now, and let's see what we get to.
Everyone should be coming back now. Is Santiago back? I don't see him.
Yeah. No,
Oh, yeah,
I went
there
to
he is.
get
There he
a
is.
glass of water.
No worries.
And
Good.
lost between the rooms.
Okay, so I think I don't really need to say much. I will hand it over back to Santiago and Yoav to hear a bit about what occurred.
So I suggest we start with group number one. They show us what they did and we will discuss it, right? I think this.
Yep.
Yeah. So group number one.
Whatever you have.
Whatever you have, even if it's nothing.
Did you see my slide?
Yes.
Yeah. So here we have tried to plot it from the 2012 to the 2022, the chlorophyll data, particulate organic carbon data, and the SST. So in
Where?
this,
What part
am I
of the
audible?
world?
It's the global ocean data.
Wow.
Oh, wow.
Yes. So here we can see the chlorophyll and POC is the directly proportioning, and from the data we can see that chlorophyll concentration and POC concentration is decreasing over a decade, but the SST is not correlating with this chlorophyll and the POC. Also, cyanobacteria concentration is increasing over a time from 2005 to the 15, where we can see we don't find any peak in the POC or the chlorophyll.
Very
Also,
nice.
yeah.
Okay, continue.
Also, from one parameter, so some of our group has suggested to. So I plot the nitrate versus cyanobacteria monthly data. So in this, the nitrate concentration and the cyanobacteria concentration is anti-correlating because the R value is 0.3. So we are not much sure about how this data is taken out. I mean, from the observation or the measurements. But yes, we got this.
So this is very nice. A few things I want to say. First of all, and I think for next time maybe we need to change a little bit the title of the workshop, because probably at least some of the data used is not it's okay, right? It's just a comment. It's not remote sensing data, but in these portals they also have models and in situ measurements. So I'm guessing at least some of the data is not from satellite. This is just something to bear in mind. So and there are many things to see. First of all, maybe there is obviously the annual cycle is always a strong signal when we look at the Earth system products. Just a word of caution, and then I will finish for the moment. Global data is always very tricky because you often mix orange and apples, right? This is how you say it, because so this is just something to bear in mind. But do you have any conclusions or other things that you want to say?
Not much, because I see just trend between the chlorophyll and the POC. Otherwise, that nitrate measurement the number was very less, 121. So that's why maybe that doesn't correlate with the cyanobacteria.
Or maybe we can conclude that more the cyanobacteria, more the utilization of nitrate nutrients. That's why we might have got the negative correlation. We are not sure whether it's significantly correlated or not. That can be checked. But if it is, then we can conclude that consumption of nitrate might have resulted in increase in the concentration of cyanobacteria over the period of time.
Okay. Santiago, any comments?
No, well, I mean, I guess I'm curious myself. I'm an atmospheric person, so anything about ocean is just very new to me. But there are a couple of general comments. You're looking at global data. Remember, you're making an average northern, the summer is summer in one hemisphere and it's winter in the other one, and vice versa. You're putting all together, and then there is the issue of sampling. Remember the northern hemisphere, you have the land mass is very different than the southern hemisphere, and observing the ocean, particularly in the high latitudes, is very difficult. So your sampling is very poor. So as I mentioned in one of the group, I mean, you are playing with monthly data, but the actual number of days contributing to that monthly average is 1 or 2 days. In the Southern Ocean, that's very possible. And we're talking about satellite measurements for 1 day, and maybe that wasn't a great day. Maybe that day the phytoplankton wasn't. So be mindful of those things. That's my comment. So anyways, I think it was an interesting trend. For example, that's something that gets you intrigued, and I guess it's a starting point. That's how you start, and then you dig deeper.
I agree. Okay, thanks a lot.
Thank you for this information.
Thank you. No, thanks. It was really nice.
Yeah, exactly. That's what we want.
The idea is to explore the data and to try to get some ideas on how to use it and how to compare. So this is exactly the idea. It's very nice. Let's go to group number 2.
Yep. So we mostly spent our time talking and trying to infer what we might observe from our results. We don't have much to actually show. But what we do have is this kind of interesting map, which was only for a month, and the idea would be to run several years of data to do trend analysis, like temps and slopes or these kind of things. So what is shown here is the Pearson's correlation between the methyl sulfide, chlorophyll alpha measured by the aqua satellite, so from infrared, and MSA, which is one of the oxidation products from DMS, which is emitted by phytoplankton. So we can see this nice correlation about the Atlantic, so which is kind of like a telltale sign of phytoplankton activity.
Now we see an image of the Mediterranean.
Yeah, so there's the Mediterranean, and on the upper left corner, you can see this huge reddish area. But over the Mediterranean, we do not see much activity, activity from phytoplankton.
Okay.
So yeah, we looked at many other parameters, but we didn't manage to really infer any like straightforward conclusion, because I think that all of this is somewhere a bit qualitative, I'd say, because we don't have like the uncertainties from the satellite measurements. We are not sure like if this is really rigorous and accurate. So, and yeah, obviously we need a lot of like background knowledge to really try to find like relevant things to compare. Like, as you said, we cannot compare April and bananas, but yeah, it was an interesting exercise, especially like since we saw that on the NASA website, we can have a look at all the events which took place like all around the world, and eventually try to infer any things from the time series, like if there are any volcanic activities or whatever. It would be nice to see that in the time series for sulfate or whatever. Yeah, it's a bit messy. Sorry, we don't have.
No, but as I said, and I think Santiago will agree with me, the main goal is for people who are not used to using this data to make you feel comfortable of just exploring it. We're not expecting anything qualitative with this 1 hour exercise, but the idea is that the data is there, it is waiting for us to use it and to just explore. And I think this was a nice example on how to do it. So it was a nice example.
Yeah, this was very exciting. There's lots to do. Yeah, for sure.
Good. Thank you. Number 3.
I'm not sure what group we are, but could I just ask the previous group, what did you show there with the volcano? What was that? I didn't get what we were looking at.
Yeah, so on the NASA website, you have all the events of like storms or volcanoes happening all the time, and you can extract time series from satellite and see if this somehow, if you can pinpoint any events from volcanic eruptions or whatever. But
Okay,
we
but
didn't do any analysis for that.
no, but
Yeah.
then the orange dots, what were those like on the map that you showed? Or that different events or?
These were the sulfates in Dobson units, like colored in orange. I think.
Okay,
Yeah.
okay.
Yeah.
Yeah. Thank you. Sorry.
No, it's good.
And I think we are group 3, and I think you have the slides. Is that correct?
I think we all have the slides, but
right.
yes,
you
but
should-
maybe we can help out then.
Yeah, I think we need to help each other out here.
Okay, but I can share the slides and then we can yeah.
Yeah, thanks.
I think our group did more of a visual analyze. None of us have really worked that much with satellite data, or at least the ones that were participating. So yeah, but we built a story.
At least we had a lot of ideas and wanted to check out a lot of different parameters, and I'm not really sure what we ended up with in the end. But
so we started off with a lot of looking at flooding data and weather events, and I found the SST anomaly in global SST anomaly data, and then we thought, oh, maybe it's a La Nina year. Yes, it is. So here we go.
That's good. Yeah.
Nice. You see, just by looking at the data, you find you get signature to worldwide to phenomena. It's beautiful. I really like it.
Okay, nice. Yeah. So this is the beauty of satellite data then. No, but then we talked about, so I mean, here you can see, can you see my cursor?
Yes.
Yeah. So you can see this up, like a blob of colder water upwelling coming here and spreading around the equator, which is typical for a La Nina year. And we confirmed it by Google. But then we also thought how to confirm it, not by Google, but by other data. So we had some suggestions on like atmospheric pressure, but we didn't know how to find that on satellite data. And then, so one suggestion was to look at precipitation rates. So this is the same time period really, but precipitation. But then we also had this discussion on precipitation. I mean, it's like on anomaly temperature, you can look at that on a daily basis perhaps, but precipitation on a daily basis doesn't necessarily say anything about these kind of events.
Right.
But then
there is another difference, that anomaly takes into account the history or the entire and if you have a daily image, then you don't have just the daily data, then you don't have reference to where it is with respect to other years. So it's very different data in a sense.
Yeah, very true. But then moving on, maybe you can take over. This is the same data, right? Like similar data, but we also did it in Giovanni, because yeah.
Good.
Good.
Very nice.
You're muted if you're talking. I don't know if you realize it.
Sorry, I had some troubles unmuting myself for whatever reason. Yeah, this is again just SST, but from Giovanni. So it's it's averaged over the 2 last years, which were both like, or when is it averaged over? Actually, 2020 January to 2021 December. So it's like La Nina-ish conditions, but it's not an anomaly. It's just the average SST. And then the idea was to correlate that with a lot of other variables, but then time ran out. So this is the average map of eastward flux, and I haven't actually really had time to look at it. I just threw it into the document, and I was actually aiming for a different water vapor variable to sort of see if the higher SST water vapor, and I don't really. I have nothing to say about this, if I'm to be honest. There was really no time to evaluate the maps we created.
No, but this is exactly what we expected, this exploration of data and trying to look what fits what. Beautiful. I really like it.
I guess one comment, a general comment I have is that be mindful when you use model data and satellite data. A lot of models, they use satellite data to do the runs. They assimilate the model data. So if you end up comparing the model with satellite, you have to remember, you have to have information on what the model ingests as satellite data, because otherwise you are going to have a fake correlation.
This is correct.
That's tricky too when you're not used to working with satellite data. I said that to the group, like it's hard to know what's a sensor and what's a model, because you have these fancy names and everything. So it's like, okay.
Well, I mean, and that's the thing I mentioned to one of the groups is, the way we do science now is getting so interdisciplinary that you need to work in groups with people who have different set of skills, and having a remote sensing person in your group is someone who will facilitate that interpretation. Yeah, that's basically the trend, I would say. Yeah.
Very nice.
Yes, I think that was no, this is water vapor as well. Okay, yeah. This was it.
Okay.
This is water vapor from worldview, but I'm not sure if this is average or if this is also daily.
No, that's daily.
This is also daily.
Yeah.
Coming in from last minute. So yeah. Thank you for listening.
Work in progress.
Yes.
Yes, Lisa. Lisa wants to say something.
I just had a thought. It's not combining atmosphere and ocean data, but a way to confirm your La Nina interpretation could have been to look at sea surface height anomalies.
Yes.
Maybe.
Yeah.
But that's an oceanographer talking. Yeah.
No, but that's very true. Yeah.
I think one thing I like, and I think I allow myself to say it's a bit underused, is the spatial structures, and the fact that you identify the spatial signature and try to interpret this. It is I really like it. It's often it's much harder to quantify. I mean, in general, it's harder to quantify because in time series, we have all the statistical tools are much easier to apply, but identifying a signature in the spatial signature is very nice. Is I think very meaningful, and we find nice results. So I like it. Very nice, very nice. Last group. So now it's easy. It's the only group that didn't talk.
Yes. I can just share my screen.
Yes.
Okay. So we plotted the data on the Indian Ocean and tried to draw a story on how the cyanobacterial abundance, like how the different factors influence the cyanobacterial abundance. And we plotted the dust deposition, chlorophyll A, sea surface temperature, and also the nitrate and the cyanobacterial, like how it's influencing. So here, like what we see, like in the chlorophyll A concentration, in April, there is the the lowest, whereas in terms of iron, it's just started increasing. And this iron is one of the more important factors for the cyanobacterial growth. And here also the dust deposition, in the dust deposition also that includes the iron that helps in the cyanobacterial growth. So here is also in April, like we targeted the very austral summer from the southern Indian Ocean. And if you also look at the sea surface temperature is also helping, like in this peak region from mainly the austral summertime, particularly if we focus on the April time period. So these are all data influencing that, and here the nitrate, so they are utilizing the nitrate, so the nitrate value is very low in the April. So, and here the cyanobacterial, like the abundance monthly, so it's April, that's becoming like it's growing in the like log phase. So from this all I can say like the higher sea surface temperature and dust deposition influencing the cyanobacterial abundance. This is the main output that I try to draw from this, all the data that I plotted, the monthly data over the 2 years.
Nice. Very nice. I think what you show us is an example to one of the biggest challenges we have, and it is to decouple the different effects. And this is especially mostly difficult when we're talking about seasonal signals, where I mean signals where you have a strong seasonal signature, and decoupling. So saying what is the cause and what is the effect is very, very challenging. It's very nice. Often the first thing to do would be to do exactly what you did. Look at the seasonal cycle and see how things are related on this scale, and then you look for anomalies, you look for different variability patterns to identify actual causality or to get some mechanistic understanding. Because everything, not every, in Earth system, in the Earth system, almost everything is seasonal. So you will always have, when you look, Lisa is laughing, but if you think of ice, for example, and everything is seasonal. So largely speaking, so this is the first. So, but I'm not saying it's wrong. I'm just saying this is always the first step, and then to go beyond, then you need to either have a model or to look for anomalies or more into high resolution, etc. But this is very nice.
Thank you. Because I earlier never used Giovanni or anything. No. And I checked the factors that I found, it's perfectly correlate like what we know in terms of the biogeochemical perspective.
No, it's a very nice story. Just to decouple, to say what is the cause and what is the effect is very difficult. But right. This is what we expected you to do. It's exactly what we expected. It's very nice. I like it. Santiago, do you want to add something?
No, not really, because it was mostly ocean stuff. So I guess, but no, but one thing that always intrigues me, I mean, it's like, yeah, I agree. Just look at the seasonal part, but in the last plot you had suddenly, I think it was in January, there was like a little peak. It was the trend was like going down and then going up, but there was a little blip. And that's what always gets me curious. What's doing this? And that's when you start to dig in and look for higher resolution. Maybe you just, like I said, maybe it was just 1 day of measurement, because also you were including from the 35 south to all the way to 65 north or something. It was a very wide range of latitudes. So you need to start to dig in and decouple things. Yeah.
Very nice.
Thank you.
So I really liked what you did on all the groups. It was really, really nice. And if anyone has comments or questions, good. So Krista, you want to wrap it up?
So I just asked Paul if he could make a poll, but I'm not sure if it works, because I'm very curious to know how many of you guys learned something about handling this data, because many of you said that you never played with this data before, but maybe we can't get to it. I'm thinking too late. Oh, poll is on the way, he says. So obviously an anonymous poll is totally, or no, this was Becky. I'm just curious because I think it's short, these practicals online, but I think it's a nice opportunity. Okay, here you go. So I would like to hear what you people say. I think it's a nice opportunity to get your hands on something and try it. And I wonder.
Yeah, I think this is the first time we do something like this online, and it was a very short window. So
Yeah. I mean, it's very challenging for the practical leaders, and it's big data sets, and it's a lot. And so I'm just curious. Okay, so how do we know when it's over? Do I decide when it's over, or?
I think you had an option to close it. You can probably.
Okay, so Paul is monitoring.
Yeah.
Or maybe it's already good enough, 60%. So apparently there are just a few people that didn't answer it. So it's all anonymous. We don't know who you are. Just if you don't mind, answer it. So I'm not answering it, obviously. So I don't know how you gauge the number. So there are something like 9 people sitting in the room that are not going to answer this question. Okay. So it looks like so far that it's half-half. Some people did work with the data, some people didn't. So satellite data, and it looks like most people learned something new in this session. So yay, I'm happy to hear it. All right, thank you for that. Yeah, I just think it's a nice opportunity to get your hands on something that maybe you would not have touched if nobody made you do it. So I hope that it can be useful to you in the future. And remember, please, that there are these links. This assignment is on the Whova. You can always have access to the links for the future and to the recording of the plenaries.
Yeah, Krista, just you remind me one more thing. If any one of you wants to start using a little bit remote sensing, feel free to contact me or Santiago. We will be happy to help with this. It's very simple and it's
no
very
problem.
useful. So feel free. This is the message. Sorry.
Great. So we're going to take a short break now. I think it's 5 minutes. So we want to come back 5 minutes after the hour. But there was one student that asked for a change in the lineup. So everybody can go take their break, but I think it was Mariana asked for a change in the lineup. If you don't mind sticking around and maybe talking to Paul and I and Becky, that would be great. And see everyone back 5 minutes after the hour.
Okay.
So Paul, I got a message on Whova.
Yeah.
That Mariana wants to go first, basically.
No problem at all. We can make that happen.
Mariana, are you there? It would be good if you spoke up. And...
Yes.
Okay.
Sorry.
Yeah. Good. No problem. No problem. Just want to make sure I got that right because I just read it really fast. So is it okay if we put you first then?
Yes, for me it would be better because I have a class in 15 minutes.
Yeah. Okay.
Okay. Yeah, we will shift you to the top of the order. Thank you.
Thank you.
Okay. Take a few minutes break.
Sure. See you later.
Hey, Tom, you got a costume change. You're muted. And we're recording, by the way, so just know that. You're muted.
I didn't hear what you said, sorry.
Oh, I said you have a costume change.
Oh,
looks
background,
like,
jumper
no,
off.
your shirt, I think. Yeah, okay, jumper off, yeah.
Don't go any later in the evening, could get crazy.
Okay,
oh, we're being recorded. I've got to stop.
Yeah, exactly. Okay, so we're coming back in 5 minutes or yeah, so see you.
Perfect. Okay, I'll see you in a bit.
So it's 5 minutes after the hour, at least according to my clock. All right, so I got the thumbs up. So here we are back for the last session of the day, the lightning rounds. So it's time for the students to give their 1 minute pitches of their posters. There's a lineup. I think we have a slide for that, but it's been switched around because one of the students requested a change in the lineup. So the way this works is your poster will come up. You will give your 1-minute pitch, and then you will see this intermediate slide come up when your time is up, and Paul will write time in the chat. And then you will have to exit the stage and the next one is up. Okay, so I'm ready. Let's do it.
Hello, everyone. My name is Mariana. I will present my scientific idea for my PhD. This idea is very incipient. I haven't enough time to look deep into it because I'm just in the beginning of my studies. Actually, I thought I wouldn't be accepting SOLAS because of that. Oh, I was just starting, but it's the same. I have a lot of curiosity. So the idea is to look on mesoscale eddies, and we pick, myself and my advisor, we choose the mesoscale eddies from Brazil-Venus confluence. So the idea is to understand how warm core mesoscale eddies affect the atmosphere above it. I mean, they could enhance any extratropical cyclone, for example, or no, or we need a more context, more information to understand that. So we decided to track these eddies to understand the path and understand how this displacement changes the atmosphere. And to do so, we will use an automatic algorithm and use altimetry. Oh my God, sorry. And Rihanna's data set.
Okay, thank you. Next.
Thank you.
We can't hear you, Shada. We can't hear you. Still nothing. I'm sorry. You're not muted either. I don't know.
Can you hear me now?
Yes, 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 yes.
Okay, fine. So my name is Samira Musidrisa from Cape Verde. This poster summarizes my master project on aerosol optical properties characterization over São Vicente during the ASCOS campaign. So the purpose is to analyze the different type of aerosol present in the atmosphere column of São Vicente by using a ground-based LIDAR data. In this context, we use the AEX techniques of aerosol species separation that we apply on different measurement cases. The first case is the 10th of September, where we found a typical dust, a typical Saharan dust in the elevated aerosol layer and a marine species in the marine boundary layer. For the second case, it is on 24 September. There also we got a Saharan dust in the higher layer and a volcanosulfate particle mixed with the marine aerosol in the marine boundary layer. So if you want to have to know more about the methodology that we use and the actually the result obtained, please have a look on my poster. Thank you, or just sending me a message.
Great, thank you. Next. Are we having a hard time?
Hey, Krista, we've had to skip a couple of speakers
Okay.
really last minute. So Beth Cowling, you are up next. Please do unmute and get going.
Hi. Hi, I'm Beth, based at Heriot-Watt University in Edinburgh in the UK. My research is investigating how organic matter produced by tropical rainforests can impact the composition of surfactants in the surface ocean, which we know from the talks earlier can affect air-sea gas exchange. So the area I'm looking at is the Essequibo River in Guyana in South America. I collected water samples from the river and coastal region and used optical analysis to distinguish between the coloured and invisible components of dissolved organic carbon, or DOC. My findings so far indicate that the surface microlayer, the SML, is enriched in DOC throughout the river and estuary with a distinct difference in composition, and invisible DOC that's potentially originating from the rainforest is present in the ocean SML, meaning it could be surface active and therefore contributing to reducing carbon dioxide exchange. Thank you.
Great, thanks. Okay, next.
Okay, hello everyone. I'm Yuanxu Dong from the University of East Anglia and the Plenary Marine Laboratory. I'm now a third-year PhD student. So I'm working on the eddy covariance air-sea CO2 flux measurements. So this poster shows our recent measurements in the Southern Ocean. So we generally use the bulk method using the air-sea CO2 fuel gas state difference measurements combined with the wind speed dependent gas transfer velocity to calculate the flux. But this is actually an indirect flux. So we use the eddy covariance method to measure the direct flux. So the first figure shows the cruises in the Southern Ocean. We collected around 7 cruises from 2019 to 2020. And if you look at the last figure, it shows the gas transfer velocity derived from this eddy covariant flux versus the wind speed. And I compare it with the widely used gas transfer velocity from Ho and Vannihoff. Looks like they agree very well, but at the low wind speed and high wind speed, it do show some difference. So this difference is very interesting. I'm going to look at it in the next few months. Thank you very much.
Great, thank you. Okay. Next.
Hi everyone, I'm Brando Yuri Damini. I'm a master's student at the University of the Rio Grande, Brazil, and my work is titled Antarctic Slope Front Bifurcation Eddy, a stationary feature control the CO2 dynamics in the North Antarctic Peninsula. Well, it's known that the Southern Ocean alone contributes around 43% of anthropogenic CO2 uptake over the past century concerning the global oceans. Therefore, the Southern Ocean is a pivotal regional to understand the environmental drivers that regulate sea air CO2 exchanges, especially considering the climate-driving changes affecting the region. In addition, recent studies have shown the CO2 fluxes could be modulated by the eddies in the Southern Ocean. Thus, here we study an anticyclonic stationary eddy located in the North Antarctic Peninsula formed due to horizontal strong current shear associated with the bifurcation of the Antarctic slope front and Bransfield current upon reaching the topographical rise in the region. Thus, using zero physical, chemical, and biological measurements like temperature, salinity, dissolved oxygen, total alkalinity, and dissolved inorganic carbon obtained along this edge, and preliminary results indicate by the surface map that the observed edge has a colder and salty core. In addition, the edge should be a regional source of CO2 during crested dissolved inorganic carbon inside the edge. Well, if there are any questions, please contact me by email, and thank you very much.
Perfect, thank you. Okay, we have one last one, and I think this was pre-recorded because the person could not come. Will you load that? Yeah.
I'm going to briefly introduce the Reef Restoration and Adaptation Program, or more specifically, the Cooling and Shading Subprogram. This project is a collaboration of several Australian universities and organizations, but we also have some international partners involved, with the aim to restore and preserve the Great Barrier Reef. And the Cooling and Shading team is trying to achieve this by applying marine cloud brightening and fogging interventions, which I'm going to show you in the next 2 short videos. This one shows the sprayer, which is used for the marine cloud brightening. Seawater is pumped under high pressure into the sprayer, where it gets dispersed in a large number of nozzles into fine droplets. And these droplets rise to cloud base and can act there as cloud condensation nuclei. Another method, which uses the same technique, produces an artificial fog over the water surface area. You can see this in this video. And both methods should provide shading over the reef to prevent the corals from dying.
Okay, then I think we made it through.
Hi there, Krista. Sorry to mess around, but somebody's internet dropped out and they are now back in.
Oh, good.
So I believe that's Margaret. If you're ready to come and present, we'll get your poster up and running.
And Latif also has his hand up. I just want to say.
Okay, hello.
So you will also present now, right?
Yes, definitely. Yes,
okay,
absolutely.
so but after Margaret, or how does it go, Paul?
Okay.
Let's go for Margaret, then Latif, and then finally Philippa. Philippa, that's my mistake. I didn't spot that you were here in the meeting. So I will start with Margaret, then Latif, and then to Philippa. Thank you.
Great, yay, okay. Did we lose Margaret again? Oh.
I think we have, yes, unfortunately. So we'll keep an eye out. If they come back, we'll line them up.
Okay.
Latif, we are ready for you to go. We don't have your poster because it wasn't uploaded to Hoover. Do you have your poster that you're able to share yourself?
Actually, my poster is uploaded on the Hoover, so I don't know why you don't have a copy of it. Unfortunately, I'm using my mobile phone now, so I'm not able to show it.
Okay.
But do you, sorry, can I break in here? Do you have something to say about your work for 1 minute? I mean, you're also welcome to present your work for 1 minute if you have something to say.
Okay, I can quickly maybe go through it. May I?
Yes, please do, yes.
Okay, thank you. Actually, it's a project I'm about to start. I'm just starting, so I've not really done anything much on it, so to say. So it's a project along the Gulf of Guinea. The Gulf of Guinea is located in West Africa, and some sections of this Gulf of Guinea are experiencing coastal flooding. So, and to understand the flooding in this specific area I chose, a key important component you need is bathymetry. So unfortunately, there's not really been any work on bathymetry on that region. So my work usually is getting this bathymetry data, which could be fed into a model that could describe the coastal flooding in this area. So unfortunately, like I said, I don't have any results, but if I keep on working on it, so to say, so soon I'll be going for the field work and trying to do the survey and get data, then start the analysis. Thank you.
Thank you. I posted your description.
Please, can I come in? Sorry.
Oh, great.
Okay.
She's back.
Yeah, please. Can I share now? Can I share my screen now?
Paul, how do you want to do it? Do you want to put up Margaret? You don't have to share your screen, Margaret. You can just talk.
Okay.
Yeah, I think
all right.
Philippa, accept our apologies. Margaret's here and your internet is not very good. So Margaret, if you go now,
Yeah.
Philippa will be ready for you afterwards.
Yeah, that's fine. Yeah, that's okay. Please, thank you. Is the internet that is on and off. So as you can see, the title is we're trying to this is part of my PhD work, which I just concluded last December in the Southern Ocean. That is south of 34 degrees. I hope you're hearing me. Yeah. Okay. So we're just trying to bridge the gap, provide more data, especially in the winter season where the data is carbonate data are really like a few or very sparse. So this is just like I was asking the question during the lecture. I said we discovered that just within the period of 2 weeks in the subtropical zone, we discovered that the carbon flux where we're going, the sampling, we discovered that there was some outflux of carbon dioxide into the atmosphere, and where we're coming, we discovered that we had a carbon sink, and yet we have the same temperature, the same temperature and DIC so to say. So we are still looking at the process, and I got some clip from the lecture, just as the two lecturers gave me some clue about the DIC ratio and maybe influx of maybe another water mass. Then we are still looking at the data, and our RF value also shows that we are having less buffer capacity of the sound. That's just the surface analysis for now. So thank you. Nothing much yet. We are yet to do the process analysis, maybe drivers. Thank you so much.
Okay, thank you. Perfect. Perfect timing. Okay,
I hope you heard me. I hope you heard me well.
We did. We did.
Because my internet was all right. All right. Okay. Thank you so much. Yeah.
Okay, hi everyone. I'm Philippa Rickard. I'm a postdoc at Newcastle University in the UK, and my research focuses on surfactants in the SML. We know that surfactant activity is a control of gas transfer velocity at the ocean basin scale, but we don't know a whole lot about them in freshwater systems. So this project is quantifying seasonal surfactant variability at a freshwater lake in Sweden with gas flux estimates via eddy covariance. Now, the fieldwork for this project finished just a couple of weeks ago, so the data are still being scrutinized, but this poster does show some preliminary data of surfactant activity against wind speed, which shows a difference in total surfactant behavior between summer and winter. So we are looking at dissolved organic matter composition as well to try and pin down why this difference is occurring. So hopefully I'll be able to tell you in about a month, but if anyone wants to chat about it in the meantime, let me know. Thanks.
All right, perfect. We have gone through it all. So we still have about 7 minutes remaining. If you guys have any questions for each other about your posters, now would be a great time to ask or express interest. Anyone want to raise the hands or ask a question? I will wait a minute. Okay, no questions. So I just realized actually that I think Margaret was on. So right, sorry, Paul just wrote me. Good point. If any of the lecturers have questions to the posters, this would also be a good time too. So everyone is welcome to ask questions or make discussion points with the posters. But I just want to say, okay, I wait for hands to raise, and while I wait for hands to raise, I keep on talking. So I just realized, oh, Tom raised his hand. Good. Go ahead, Tom.
Hi, my internet's decided it's going to cause problems, but hopefully you can hear me. I had a question. It was about the stationary eddies. I wondered, I hadn't heard of these before, and I wondered how frequent they are and where they are in the ocean, or were they just in the Antarctic? Or can't remember the speaker. Sorry, that's not really.
Yeah, I'm looking now.
It's Mariana.
Ah, then she left. She left, I think, because she had to. But you should write the question to her in her poster. That would be great.
Okay, thank you.
I'll answer that question. Tom, I think they're topographically controlled. Could you hear me?
Yeah. Hey, hi, Eric. I did not know you were there.
I'm there.
Snuck in
Lurking.
there.
I think they're topographically controlled. So there's some big features in the Southern Ocean that spawn eddies in a consistent and steady position.
Huh. And would they? So does that mean you'd get them somewhere else as well if you have the right topography? Yeah. I assume.
Yes, anywhere you have a rise in the middle of a big current, you probably spawn some sort of eddy, but I don't know if there's a lot of them or not.
Thanks, Eric. Nice to see you.
That's funny. I didn't even know he was there. Great. So any other hands or questions to the posters? I noticed there's a decent amount of overlap between some of the topics, and I hope that you guys will talk to each other. Even if you don't want to do it now in front of all of us, please do it on Whova, because I think you have the potential to learn from each other, or at least have collaborations and discussions into the future. I can say that Tom and I met at the very first SOLAS summer school, and I still talk to him. So I think you should really explore your options here at the SOLAS summer school.
And vice versa. You don't just talk to me. I do talk to you.
Okay, you talk to me too. We talk to each other. Yeah. Okay, if nobody has really any questions, then I would say we can wrap it up for today. And before so I'm going to keep on talking, and if anyone wants to speak up, then please raise your hand. But I can close today by saying that tomorrow we meet, let me check the time so I make sure it's the right time. We meet at 11 o'clock UTC, and it will be on the SOLAS core theme 3 tomorrow. Yeah, and I think I just want to encourage everyone, please, to go to Whova, interact. I see that there's a lot more chats going on, a lot more community interaction. I encourage it. I think it's great. And don't forget to upload your photos for the photo contest and vote on each other's photos before, I think it's 1300 UTC on Thursday. So that's the end of the contest. So yes, and if you want to enter the poster competition, please upload something for the posters. Great. So I look forward to tomorrow. Thank you today for a great session, everyone. And yes, see you tomorrow.